Can you guys hear me? I've not seen any chat on the... Oh, okay. I thought I did something wrong. I thought I didn't set it properly. Sorry. Oh, perfect. Let's just give a couple of minutes for everybody to be here. Just a few minutes. Okay. Okay. So how are you guys doing? We are learning a lot today. Welcome. Try to get this in motion. Okay, so it's time, guys. I hope everybody's doing well. Nice to have you guys around again today. So today we are discussing about 6 to 7 chapters. It's a crash. We are going to crash a lot of things. I hope all of you are ready. I hope everybody is ready for it. So I'm going to share my screen. And we'll follow the normal protocol. Somebody is already injured. Please don't be injured. I need all of you to be active. I need everybody to be active, guys. Because I will be flying today. I'm going to be going very fast. So if you're active, it will be better for us. And one more thing. Anytime it's lagging, please just try to tell me, right? Once it's lagging. Let me see. Okay. I think we are good now. So today we are starting with the first chapter. We are going to consider chapter 1, chapter 2, chapter 3, chapter 4, chapter 5, chapter 6, chapter 7, and chapter 11. Those are the the 8 chapters we will consider today, because I know your midterm exam will be taken from these chapters. Okay. All right. For chapter 1, I'm going to move over things that won't be too necessary for you, but you should also learn it at your leisure. Remember, the most important thing you need to know is that anatomy is structure, and physiology is function. And this structure and function, as far as your college anatomy and physiology is concerned for nursing students, it's all about the biology of the human body, all right? Biology of the human body. So A and P is actually a foundation, all right? If you are very good with what we are doing right now, if you are able to assimilate properly, it will be easy for you when you start doing your pathology, and pharmacology, even your medicine and your surgery. So at the end of the day, this is the foundation. That's why we always want to start doing it as early as possible so that students will be able to understand the entire concept. For this chapter 1, it's just all about the physicals, right? This is your examining. When you're examining the structure of human body, you inspect, you palpate, you auscultate, and you percuss. All right. For the schools that uses cadaver, there's cadaveric dissection. All right. While some other some other schools, use models. All right. So this is what you do for inspection. General inspection involves overall appearance of the health of the individual. So just by citing a patient, you're already supposed to, speculate what is possibly wrong with that patient. You understand? You are already supposed to, suspect. You understand? We call it differentials. You need to have differential diagnosis at every time. Okay. Time. And how do you check that? The overall appearance of the patient, the signs of distress, facial expression, body size, grooming, and all those things. All right. So I'll just move. This is palpation. All right. These are the things that are involved in palpation when you palpate a patient. Okay. This is percussion. You are percussing. All right. You are checking for a lot of things. You want to hear the sound of different particular parts of the body to know if there is a fluid accumulation or not. Not just fluid. There are other things you want to know. All right. That's in percussion. Now we have auscultation, which you use your stethoscope. So in your exam, if they're asking you what differentiates auscultation with every other thing, it is the use of stethoscope. We want to hear the sounds of these organs moving, all right, to know if they are moving properly, all right? And there are 4 characteristics of sounds that, you can auscultate. We have the pitch, loudness, quality, and duration. This is how you are able to explain it. Like when you are in the clinic, you are discussing with you are reporting a case to your supervisor, right? Your supervisor will ask you, okay, when you did your auscultation, what did you hear? You understand? Did you hear a short, a high sound or a low sound? Stuff like that. You describe the sound. What's the pitch? Is it a high pitch or low pitch? What's the loudness? What's the quality of that sound you heard? So all these things are extremely important, and it's very, very good that you look at it. Right? Okay. So these are other parts of anatomy. You understand? The exploratory surgery, medical imaging, gross histology, histopathology, cytology, and ultrasonography. And understand? And when you're talking about comparative, most times we are talking about other animals. These are the subdisciplines like the neurophysiology, endocrinology, pathophysiology. Okay. So I'm not so much interested in this particular history. And if you are, you could look it up at your leisure. All right. You should look it up at your leisure. Yes, this is experimental design. You understand? You do sample size, controls, psychosomatic effects, experimental bias, and statistical testing. Right? All these things we do it before we even start critical evaluation. The critical evaluation is what we call peer review. Every study that you do, you pass through these protocols, and after passing through these protocols, there has to be a critical review, peer review. All right, before that study could be accepted. And that peer review is all about checking if the study is very honest, if what you are saying is true or what you discovered is true, how objective you were during the study, the quality of the science. All right, very, very important. So we have a scientific facts, that's information that can be verified independently, and we have laws. All right, laws are just generalization about a certain behavior. Very, very important. Then we have theory. Theory is just a statement or set of statements that derive from facts and derive from laws. And when you've derived this thing, you confirm the hypothesis. That's when you now start saying this is theory. So you need to be able to differentiate between fact, law, and theory. Extremely important. This one, I'm going to jump this. This is evolution, artificial selection, and others. I'm going to jump this and see if there is something you need to know. Okay, yes, you need to know the hierarchy of human life, all right? The hierarchy of complexity of humans, all right? So you can start from the smallest compounds, which is or molecules, which is atoms. So atoms are the smallest, but when you group atoms together, they make up molecules, and group of molecules make up organelles. Organelles make up what? Cells. Cells make up what? Tissues. Tissues make up organs. Organs make up organ system, and organ system make up the organism itself. They can give you this diagram and label this A, B, C, D, E, F, G. All right, and they ask you, okay, this particular one, what does it signify? This is organ system. This is organelle. This is atom, the first one, followed by molecule, macromolecule, molecule, organelle, cell, tissue, organ system, and the organism. You need to be able to understand this. They may not give you this as a diagram. They may give you as an arrow arrangement. So you need to understand it. So I need you to look at it properly and be able to understand it. Okay. Let me see. So this is talking about variation. Now, when you are talking about physiology, that's human function of human organism. These are different functions or characteristics of life. All right. Organization, cellular composition, metabolism, responsiveness, movement, homeostasis, development, reproduction, and evolution. All these things are just characteristics of living things, and which we are able to differentiate living things from what? Non-living things. This is a summary of the characteristics, which is movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. I may be talking very fast, but if you can't hear me properly, please always say go, Nchebe. So if you can't hear me properly, please just type on the comment section so that I'll be able to drop my pace. Right now, I'm talking slowly because I'm just starting. Once I enter the groove, you know how it is now. I'll run away. I'll fly. All right. So we have different variations. All right. We have continuous and discontinuous variation. All right. Continuous variation is usually the one that goes this way, or it has intermediates. Like what? Like height, like skin color, like weight. All these things are what? Continuous variation. Then we have discontinuous variation. Discontinuous variation is the one that does not have intermediates. For example, some people can test PPT and it will be testless. Other people will test it and it will be what? Beta. All right. So that is discontinuous variation, or ability to roll the tongue. You know, something like. Try it. Try it. Can you roll your tongue? Can you? So some people can roll the tongue. Some people cannot do it at all. Like it's all or none law. It's either you can or you cannot. That is also a discontinuous variation. All right. So continuous variation are the ones that have intermediates, but discontinuous variation are the one that doesn't have intermediates at all. So that's all about variation. All right. So everybody is different in one way or the other. Now we have homeostasis, and homeostasis is the body's ability to dictate change. All right. And when the body dictates change, it tries to go back to normal. So it's having a constant internal environment. All right. Your internal environment being stable. So no matter how hot it is outside, your inside is constant, the same thing. No matter how hot it is outside. So what does the body do? The body knows that, okay, the outside temperature can affect the inside temperature. Then the body changes itself internally to be able to accommodate that external temperature. You understand? And still remain constant. So that's what homeostasis is. All right. Then we have something we call negative feedback. Negative feedback allows for dynamic equilibrium within a limited range around a set point. So if this is a set point, all right, if this part is going up, this part will be coming down. That's negative feedback. So as this part is coming down, all right, it's telling this part, all right, you have to come down. And if this is coming down, this one will go this way, and they maintain homeostasis. That's just, a practical way of understanding what negative feedback is about. But we'll discuss it more. All right. Now, when there is loss of that homeostasis, maybe this one is here and this guy keep going up without nothing to oppose the effect of this guy. All right. It leads to what? Death. And also that's what causes what? Illness. So loss of homeostatic control is the cause of illness or death. That's extremely important for you guys to understand. Okay. Okay, so these are some of the examples of homeostasis, common homeostasis. If you are too warm, your vessels will do what? Dilate in the skin, and sweating what begins. So you are too hot inside, your vessels dilate, you start sweating. Once you are sweating, cooling starts happening. So your body, your internal environment goes back to what? That set point. That's just what this place is talking about. And when you are cold, what happens? You start shivering. Your skin starts constricting, you start shivering. And when you start shivering, what happens? You generate heat inside. So this is what we call what? Homeostasis. All right. Okay. So you see, this is a negative feedback in thermoregulation. Room temperature, thermostat activates the furnace. The furnace starts giving out heat. Heat is outpouring. Once the heat starts getting too much, what happens? The thermostat shuts off. Everywhere starts getting cold. When it gets too cold, if it falls below around 19 degrees centigrade, the thermostat activates. It gets hot again. This is what? A negative feedback in thermoregulation. And this negative feedback is all about having this thermostat in a constant temperature, which is what? This environment in a constant temperature, which is between 19 degrees centigrade and 20 degrees centigrade. All right, that's 67 degrees Fahrenheit and 68 degrees Fahrenheit. So everything is all about this being constant, which is what? Homeostasis. And this homeostasis, what is the mechanism it's using to happen? For negative feedback. Now, there is negative feedback, there is positive feedback. All right? So that's exactly what they explained here. Okay. So these are examples of negative feedback. At the end of the day, vasoconstriction, vasodilation. If you are too hot, vasodilation happen, you get bang. If you are too cold, vasoconstriction happen, you start shivering, and your temperature goes up. So everything is all about being in this fixed point, which is at 7.0 degrees centigrade. This is normal set point of the body. Once it's above it, you are hot. Vasodilation, you cool down, you come back to 37. All right? So this is negative feedback in thermoregulation, leading to what? Leading to homeostasis. Extremely important. So these are other forms of negative feedback that you need to understand. All right? You rise from the bed. This is a hemostatic compensation for a postural change in blood pressure. Because immediately you stand from bed, what happens? Blood drains from your upper body, creating what? Hemostatic imbalance. Immediately this hemostatic imbalance is created, your baroreceptor senses this change. And once the baroreceptor senses this change, your blood pressure falls down. Pew! Immediately your blood pressure falls down. Pew! What happened? The baroreceptor sends signals to the cardiac center of the brain. That signal will tell the heart, oh yeah, you have to move a bit faster because the blood pressure has fallen down. And immediately the heart starts beating faster, the blood pressure is restored to normal. So you don't even notice this. In that immediately you stand up, your blood pressure drops. Why? Because the heart immediately corrects it by sending information to the baroreceptors, and your blood pressure is restored to what? Normal. All right? This is what we call homeostatic what? Compensation. It must be normal. If it's not normal, then you are sick. For example, there are some people that will stand up and their blood pressure drops, but this baroreceptor will not fire. It will not tell the brain that the blood pressure has dropped. Do you get? It won't tell the brain that the blood pressure has dropped down. Because if it tells the brain will send to the cardiac center and it will increase what? The blood pressure. So some people, it won't tell them. So what happens? You see the person, immediately he stands up from the chair, he starts feeling dizzy. And some of them even fall. That's what we call orthostatic what? Hypotension. Some people, they stand up and it rises. Orthostatic hypertension. So, like, as I was telling you, any change from the normal, that's a homeostatic mechanism, is known as what? Pathology. So you have to understand it. And remember, the idea why we are doing this is because we want to understand what? Physiology, which is normal. We are trying to use the normal to understand the what? Abnormal. I'm saying this because this part is extremely important for you to understand. Now, I just mentioned the baroreceptors, all right? They are just nerves that are found around arteries, all right? Some of them are near the heart, some of them are in the brainstem. If you look at it, this is the baroreceptors, this is this, baroreceptors, all right? This is in the heart, this one is close to the brainstem. Extremely important that you understand this. So baroreceptors changes. There's another one we call what? Chemoreceptors. This chemoreceptor changes what? Change in chemical. When you're acidic, your blood pressure, you have high acid in your blood, it detects it. Or when the acidity, the pH of your blood is too high, that's alkalinity, all right? It detects it. That's for chemoreceptors. But when you talk about fluid, that's usually the blood. We talk about what? Baroreceptors, all right? So that's the thing. Okay. Now, you remember the brain is integrated center, all right? We have something we call the effectors, which we also learn in muscles, all right? These are cell or organ that carries information to the final corrective action to restore what? Homeostasis. That's what we call effectors. Extremely important that you understand. So these are positive feedback mechanisms, all right? What is positive feedback? Positive feedback is usually, when when something is happening, all right, becomes a change, and that change will cause that thing that happened to keep happening more. That's amplification. I'm just looking for a simpler way for you to understand it. It's called what? Amplification. Make it happen more. All right? You are bleeding. Instead of the bleeding to stop, it will increase. That is what positive feedback mechanism. All right? So self-amplifying cycle leads to greater change in the same direction. Feedback loop is repeated. You understand? Change produces what? More change. Negative feedback, change produces what? Less change. Negative feedback wants to stop the problem, but positive feedback wants to increase what? The problem. I need you guys to understand it that way. What are examples of positive feedback? You talk about childbirth, blood clotting, protein digestion, fever, and generation of nerve signals. Look at childbirth, for example. When somebody is contracting, all right, you know now how it is. For some of you that are already mothers, you know how that contraction happens. Very painful, caused by oxytocin. You will see the person shouting, ah, ah. Immediately that contraction starts, that contraction will lead to more oxytocin release, causing more contraction. The contraction keep increasing. Trust me, it starts small. Maybe the person, the lady will feel contraction, and it will stop. Then gradually, that contraction will keep getting higher and higher and higher and higher until it pushes out the baby. That is what positive feedback change. All right? Positive feedback. Same thing with blood clotting. So what you need to know is know the examples of positive feedback. There are very few positive feedbacks in the body. Know the examples. All right? Same thing with blood clotting. Yes, cause and effect. Thank you. Cause and effect. You're correct. All right? So look at it. You see how it works? Once the head of the baby, the head of the fetus hits this particular place, hits the cervix, what happens? It will send nerve impulses from the cervix to the brain. Now, the brain stimulates pituitary to secrete oxytocin. Oxytocin stimulates uterine contraction and pushes the fetus toward cervix. This is a positive feedback. All right? Now, more oxytocin keep coming out. In contraction, keep increasing until that contraction helps to push out the baby and everything automatically stops. That is positive feedback. All right? Same thing with fever. All right? So this is fever. You can look it up later. Okay. So let me see what else you need to know for. Yeah, you need to also understand some of these informations, like some of these positionings, all right? Or plural form of things like ovary, ovaries, cortex, cortices, corpus, corpora, epididymis, and epididymides. That this is what plural form of this. Very, very important for you to know, because we'll be seeing it in some questions. And also there are some adjectival forms that you need to understand, like the brachium is the arm, brachii of the arm, stuff like that. Digiti, the little. All right. So everything is highlighted here, so you can take a screenshot if you want. Okay. Let me see if there is something else. Okay, so that would be that for chapter 1. I'll just answer very few questions for chapter 1. So I need you guys to be active. Are we still here? I keep talking. I'm not even checking if people are still here or not. Are you ready? If you are ready, put yes in the chat section. For chapter 1, yes. Let me end this so that it will not lag. Sharing. Okay. Okay. How many years do I have? Okay. I have a lot of yeses. Is there anything that yeses? Nchebe-jah. Okay. Okay, that's fine. I'm trying to open what I've prepared for you guys to. What is wrong with my network? Just a moment, please. My network is now lagging behind. Interesting. Chapter 1, right? Chapter 1. There'll be just very few things. Moments. Okay. Here we are. Finally. Okay, guys. I'm going to post just very few questions for chapter 1 because we don't have much time. Before you know it now, we've gone 3 hours. Okay. Hold on. Okay, testing the microphone. Let's test the microphone as usual. Testing the microphone. Talk to me. The study of the structure and function of cell is called dash. Testing the microphone. Testing the microphone. Testing the microphone. Let's see. How many people? Okay. So everybody, I need everybody to type the answer so that your hand will be very, very ready for what comes next. Yes. Okay. I don't know why my system is lagging. Okay, perfect. So that's cytology. Okay, so another testing the microphone. Let me see. Another testing the microphone. So I will also be reading the questions so that some people that are not here are able to, look this up at their free time. Feeling structures with your fingertip is called dash, whereas tapping on the body and listening for sounds of abnormalities is called dash. It has to be 2 answers, guys. We are still testing the microphone. Don't think this is the main thing. Come on. Please, if you don't know it, write not sure. All right. Don't keep quiet. Don't keep quiet. I need everybody to be typing. Everybody. Okay. I need 2 more people. Okay. So this is making sense. Okay. You guys are doing well. So feeling structures with your fingertip is called palpation. When you tap, it is called percussion. All right. Auscultation is the one you use your steps. Okay. So that makes sense. That's just testing the microphone anyway. Okay. Okay, so look at this question, guys. I'll give you options. It's very, very cheap. Also testing the microphone. The study of mechanism of disease is called A, neurophysiology, B, endocrinology, C, pathophysiology, D, neuroanatomy, and E, histology. What's your answer? Some people say pathology. So pathology is the disease itself. Pathophysiology is the mechanism, all right? And what you are asked is the mechanism. Okay, so the answer is pathophysiology. Yes, you're correct. All of you are correct. We are still testing the microphone, though. Bear that in mind. Okay, but I'm not going to. Let me see. Okay, look at this question. Look at this question, guys. Which process submits a scientist's idea to the critical judgment of other specialists in the field before the research is funded or published? Peer review. Okay. This is great. You guys are really activated, though. You guys are really activated. Let's see. Look at this question. No, this question is too long. I don't have this kind of. Okay, no. I'll still give. This question is very important for you to know during research. Hold on. So this is the question. I want you guys to look at it. Reduction, steps and reduction. That's the first part of the question. Then this is the completion of the equation. Okay, so two groups of people were tested to determine whether garlic lowers blood cholesterol levels. One group was given 800 mg of garlic powder daily. One group was given 800 mg of garlic powder daily for 4 months and exhibited an average 12% reduction in the blood cholesterol. The other group was not given any garlic and after 4 months averaged a 3% reduction in cholesterol. The group that was not given the garlic was the DASH group. Okay, this is great. So it's actually control. You guys are very correct. Okay, this makes sense. Chapter 1 doesn't really have so much. Trying to look at it. Okay, good. This is a very critical question, a very important question. Yes, the answer to that is control. All right. The answer to that is control. An educated speculation or a possible answer to a question is called dash. Let me see who gets this. Sorry, today I won't be giving options. So, all right. So that will be very fast. Today I won't be giving options. An educated speculation or a possible answer to a question is called dash. Okay, is that all we have? Let me see. Hypothesis. Okay, you guys are very correct. So I just want to give you guys a little differences between these things so that you'll be able to remember it. All right. We have scientific method, we have theory, we have law, we have hypothesis, we have facts. First of all, let me start with facts. Facts are actually, like something you observe, observable, proven truth, confirmed by repeated evidence. That's fact. Proven truth. and it's, you are seeing the evidence on a daily. That's fact. All right. For example, the heart pumps blood. That's fact. Fact. All right. So that's what fact is. Then when you talk about hypotheses are testable, prediction or, a guess about an outcome. Testable prediction or an educated guess about an outcome. That's what hypothesis is from what we discussed. Then when you're talking about law, it's what's constantly, how do I explain it? Okay, law is something like that constantly happens under a certain condition. It constantly happens. Like what? The law of gravity. Under a certain condition, if you throw something up, it must come down. Do you understand? That's what law is. Then when you're talking about theory are usually when, all these hypotheses or, laws has been completely well tested. So they are well tested explanation for why or how something happens. That's what theory is. So you need to be able to understand each and every one of these things so that, you'll be able to get a proper answer for it, because they can ask you one question like this in exam and, okay, good, perfect. I have something that I will know if you guys understand what I just explained. Look at this question, guys. Which of the following could contain the greatest amount of information that scientists consider to be true to the best of their knowledge? I'm not giving option. Okay, fine. Fact, law, hypothesis, equation, theory. Fact, law, hypothesis, and equation, theory. Come on, guys. We are a lot here. I'm seeing answer from only how many people here? 5 people. 10 people. Please give me more answers. I want to see more. Come on. Why is everybody saying fact? Fact is just a very limited scope. It's just a single observation. All right. The answer is theory, not fact, guys. Yes, somebody has gotten it finally. The answer is theory, because theory contains the greatest amount of information. All right. It explains why, how things work. It integrates multiple facts, laws, and observations into one framework. So the answer is not law. The answer is law just describes what happens constantly. No explanation why. But you see that why? That is why it is theory. Let me look for. Let me see if I can get a better question for this. Please, the answer to that is not fact. It's theory, guys. Not fact. Let me see. Is there a better question? No. No. So, okay. So, those are just the 2 questions for the scientific information. Look at this question, something different. And dash is composed of 2 or more tissue types, whereas dash are microscopic structures in a cell. 2 answers, guys. Two answers. Somebody says, somebody's asking me questions. Hey, you're very funny. Huh? Hey, you're cracking me up this night. And dash is composed of 2 or more tissue types, whereas dash are microscopic structures in a cell. Okay, you guys are doing well. An organ is composed of two or more tissue types. Yeah. Then, whereas organelles are microscopic structures inside a cell. That's correct. Okay, so look at this question. I'm going to post this question, then I'll post the option. Just you allow me to post the options, all right? This is the question. Then the options A, incoming is A, B, incoming. This is B, C, D, and E. So which of the following lists levels of human structure from the most complex to the simplest? From the most complex to the simplest. A, organelle, cell, tissue, organ system. B, organ system, organ, cell, tissue, organelle. D, organ system, organelle, tissue, cell, organ. D, organ system, organ, tissue, cell, organelle. E, organ system, tissue, cell, organelle. Impressive. Impressive. Yes, the answer is D. Organ system, then the organ, tissue, organelle, a tissue, cell, and organelle. Yes. So knowing the rank is very, very important. Okay, look at this question, guys. I'm going to pose this question. Please allow me to pose the answer before you start. All right. Here's the question here. Let's see. Let's see who gets this. This is A. This is B. This is C. This is D. And this is E. Let's see. Okay, you guys are doing very well. Honestly, you guys are impressive. So for people that are not sure, please, when you are not sure, type not sure. Don't refrain from typing. No. You have to attempt it. Even if your answer is different from what everybody is picking. Don't pick somebody's answer. Do you understand? That person can be wrong. And usually when you are writing your exam, you are alone. So even if you think you are failing it, that's fine. All right? So don't jump in and follow everybody. But I need you to attempt the answer as fast as possible. All right? Okay, so what you need to know is that protein, we know protein. Protein is a molecule, right? Chemical level. It's still at its chemical level. And it's the simplest, you understand? Because there are macromolecules made up of what? Amino acids. So protein starts, then after that, you talk about what? The mitochondria. Mitochondrion is at the organelle level. It keeps going up, all right? It's a structure inside the cell. Then after that, adipocytes. That's a fat cell. Adipocytes is fat cell. So from protein to mitochondria, that's molecule to organelle, then to what? Cell, right? Then after that, you talk about tissue. What is tissue? Connective tissue. Then after that, you talk about organ, which is organ, stomach. Okay, so all of you are very correct. All of you are exceptionally correct. This is the main thing this is the main thing they will be asking you. Okay, very fast, very fast. I like the way you guys are flowing very fast, but we have 50 something people here and we have 6 likes. Why? This like should be up to 30. Okay, all of the following are human organ systems except all of the following are human organ systems except the options. Pay attention for the options. All right. A, skeletal. B, endocrine. C, epidermal. D, reproductive. E, lymphatic. Okay, so all of you are very correct. The answer is epidermal. All right, it's not a part of anything. Okay. I want you to also look at this. I don't know if we mentioned it, but I think I've discussed it with you guys very fast. Look at this question. Oh boy. Hold on. Is it that long? Why is it not? Look at the question, then this is the completion. So there are 2 answers there. The change in size of the bone marrow where blood cells are produced as an infant matures is an example of dash. Whereas the transformation of blood stem cells into white blood cell is an example of what? Dash. Answer. There are 2 answers there. Do you need options? I don't think so. You guys seem very capable. Oh, you guys need option. Okay, so I was even busy trying to get another question for you guys. Okay, so option A, development and differentiation. B, growth and development. C, growth and differentiation. D, differentiation, growth. E, differentiation, development. Okay, so all of you are correct. It's growth and differentiation. All right, growth. What is the differentiation? Is specialization. All right, like some of you, most of you are nurses in general nursing. When you are done, you want to specialize. Oh, I want to go for orthopedic nursing. I want to go. I know you. You want to go for mental health. I know. It's okay. You told me now. Somebody's wondering, is he talking to me? Yes, I'm talking to you. Okay, so that's differentiation, all right? So you need to bear that in mind. Okay, I think that will be that. Now, the other question they can ask you is just homeostasis, all right? Just homeostasis. Yeah, I can see you. Okay, perfect, perfect. Very fast. Very fast. Very fast. This question, guys. This question: during exercise, one generates excess heat and the body temperature rises. As a response, blood vessels dilate in the skin, warm blood flows closer to the body surface, and heat is lost. This is an example of dash. Answer. Yeah, you're correct. Growth and differentiation. That was correct. Yeah, for the last question. But now we are in a new question. During exercise, one generates excess heat. I know all of you are exercising. One generates excess heat and the body temperature rises. As a response, blood vessels dilate in the skin, warm blood flows closer to the body surface, and heat is lost. This is an example of what? Everybody, let's go. Perfect. You guys are so good. Like, you guys are very impressive. Okay, yes. Now that's a negative feedback. You guys are so correct. You remember what negative feedback is, I believe. So I'm gonna ask you this question. Yes, you're correct. That's negative feedback. Look at this question. Look at this question, guys. Blood glucose concentration rises after a meal and stimulates the pancreas to release the hormone insulin. Insulin travels in the blood and stimulates the uptake of glucose by body cells from the bloodstream, thus reducing blood glucose concentration. This is an example of what, guys? What's the answer, guys? What's happening to everybody? I'm seeing less and less people are responding. Okay, so now the rule is 20 people has to respond for us to proceed. And if you really want us to go very fast, we are still in chapter 1. If you really want us to go very fast, then you have to talk fast. If you don't know it, write not sure. All right? Don't just keep quiet. I need to communicate with you. You understand? You know how very communicative I could be. So I really want to hear you. And the only way I can hear you is by responding to what I'm saying. So I need everybody to talk to me, please. Okay, let's go. Everybody. Okay, you guys are all correct. That is negative, also negative feedback, right? Because it's stopping. Okay, look at this question. Look at this question. When a woman is giving birth, the head of the baby pushes against her cervix and stimulates the release of the hormone oxytocin. Oxytocin travels in the blood and stimulates the uterus to contract. Labor contractions become more and more intense until the baby is expelled. This is an example of what? More people are responding now. It seems you were not sure of the other one, eh? You guys are extremely correct. Thank you so much. You guys are so correct. Positive feedback. Now look at this question. This question says, which of the following is the most likely to cause disease? Let me give you the options. Which of the following is most likely to cause disease? I hope you are listening to me. So A is positive feedback, B is negative feedback, C is homeostasis, D is equilibrium, E is irritability. Which is the most likely to cause disease? A, positive feedback, B, negative feedback, C, homeostasis, D, equilibrium, E, irritability. Yes, I like it. When you are not sure, just write not sure. Don't follow the group or whatever, the public. So if you are not sure, just say you are not sure. So that, because when you say you are not sure, it helps me to know that, okay, some people didn't get it, so maybe I have to shed a little light on it so that you remember it. Okay, keep going, keep going. Someone say homeowner or positive. Can you repeat the choices? Sure. Positive feedback A, negative feedback B, homeostasis C, equilibrium D, irritability E. Homeostasis is because of the immune system. Okay, so all of you are doing very well. The answer is actually positive feedback. Positive feedback. Someone said 911. Okay, fine. Positive feedback is the most likely to cause disease. Why? Because it makes whatever that is happening to get worse. Homeostasis is normal, gets you to be normal. You are fine. All right? You are fine. Negative feedback helps you to get back to where you used to be. But positive feedback will keep pushing you further and further. That's why you see it in things like fever. You see it in things like DIC, discontinuous intravascular coagulopathy. You understand? You see somebody bringing out blood from the nose, mouth, ear. Why? Because the platelet has, like the body is closing, and the platelet finishes. That's negative feedback. So bleeding, negative feedback. Fever, negative feedback. Childbirth, negative feedback. Trust me, childbirth is physiology, but if you have experienced childbirth, you may be mistaking it for pathology. Honestly, I've seen people experience childbirth. I have not. You understand? But I've seen people experience childbirth. And the way they cry, you get one time in the clinic, like this woman, she wanted to slap the husband. She was in labor. She was shouting, and the husband was like, "Hey, babe, I'm so sorry. Sorry. Oh, babe, take it easy." She just turned like this. She wanted to slap the man and slap me. I had to bend. I said, "Please, oh, I was not there when you guys were producing this baby, so don't include me in the anger." You understand? So positive feedback, that most likely to cause diseases. Please bear that in mind, and that should be the. Let me see if there's something else I want to push to you guys. No, so that should be the end. No, hold on. Just one last question, please. One last question. One last question. What is the meaning of this terminology? Yeah, it's positive feedback, guys. What's the meaning of this terminology? What does hypercalcemia mean, guys? It's a giveaway question, and it's the last question I'm asking here. High calcium. Perfect. You guys are so good. Okay. I'm sharing my screen now. Let's jump to the next thing on the agenda. Chapter 2. Please, I'll be very fast because chapter 2 is long. There are some chapters I'm not even going to explain. I'll just move straight to the main thing. Hold on. Is this chapter? No, this is not chapter 2. Okay. So this is chapter 2. I want to be able to see what I'm doing. Okay. So if there's something, if it's lagging, please try to tell me, all right? All right. So chapter 2 is all about biochemistry. What are we biochemistrying? What are we studying? We want to know the chemistry, carbohydrates, the fat, the protein, and the nucleic acid. All right? Because these things are important. We humans, animals, we are made up of all these things. All right? So we want to identify these things properly, starting from the smallest. All right? Then we move to the highest. These elements are the simplest form of matter to have unique chemical properties. All right? There are 24 that are very, very important for in biology. These are the 24 that are very important for biology. These are the major elements. These are lesser elements. These are trace elements. All right. If you notice, most of your what is it called? Most of your supplements are usually this lesser element and these trace elements. Those are the most supplements you take. All right. In short, you take more trace elements than every other. Why? Because it's found very little. Very, very important for you to know this fact. All right. Then we have minerals. Minerals are inorganic elements extracted from soil by plants and passed up food chain to humans. All right. Okay, I'm just jumping because you need to know your trace elements, why they are important. This is the periodic table. I have to jump. Okay, good. So you guys need to understand this extremely well, all right? This concept, because there are questions surrounding it. The number of protons is the atom's atomic number. The protons is the atomic number. Number of protons is the atom's atomic number. So this number you are seeing here now, this is an atomic number, and this is number of protons. And proton is positively charged, right? And atom's mass number is the sum of the number of protons and the neutrons. So you need to know the difference between the atomic number, the mass number, all right? Atomic mass number, very, very important. And the atomic mass is approximately equal to its mass number. So you need to understand this, all right? Because they will ask you questions surrounding it. Protons are positive, electrons are negative, neutrons are what? Neutral. So I'm not going to waste time on it, all right? So this is what we call the outermost shell. We have different shells, 1, 2, 3. This is the outermost shell. You understand shell better on. I think most of you did chemistry, so it's very, very understandable. So this is the inner shell. We call it what? The inner shell. That's the S shell, all right? It usually has 2 electrons, maximum 1, 2. Electron is negatively charged, right? 1, 2. And remember, protons are in the core, right? In the nucleus of the atom. So this is electrons. They are just outside, perambulating outside. They can't come in because here is positive. So this is negative, the first shell. This is the outer shell, all right? 1, 2, 3, 4, 5, 6. So this shell has an incomplete. It's supposed to be 2, 4, 6, 8. For you to complete the second shell, it has to be 8. All right, the first shell is 2. So if you count, they ask you now how many this thing is in this, what is the number of the outermost shell electrons? Rather, this is 1, 2, 3, 4, 5, 6. All right, it's 1, 2, 3, 4, 5, 6. So you will discuss most of them, and also know that the number of protons and electrons are the same thing. So, and remember, the number of protons is what we call what? Atomic number. All right. So meaning that if you want to calculate the atomic number of this person now, this is 1, 2, 3, 4, 5, 6, 7, what? 8. So they are 8 in number. All right. If you calculate the protons, that's the positive ions inside, you see that it's the same thing. 1, 2, 3, 4, 5, 6, 7, 8. So it's the same thing. And that is the atomic number. So if you are given this diagram and you are asked the atomic number, just count it. Count either positive or the negative, and you are good to go. All right. So that's the thing. Then this is an example. Like, for example, this is what we call planetary model of electrons, Bohr's planetary model of electrons. All right. If you look, this is carbon, this is nitrogen, this is sodium. All right. Carbon, you see, 1, 2, 3, 4, 5, 6. So atomic number of carbon is 6, but atomic mass of carbon is 12. Remember, atomic mass is number of proton and neutron. All right. Very, very important for you to know. So it's either they give you atomic mass and give you atomic number and tell you to calculate what? The neutron. Or they give you the neutron and they give you also the atomic number and tell you to calculate the what? Atomic mass. Anyone they give you, you should be able to understand it. All right. Now, this is sodium, for example. Sodium is number 11, right? So if you check it, 2 has filled the first shell. That's the 2s. This is 2s. 2 has filled the first shell. So the next shell has to be what? 8. So let's put 8 there. 1, 2, 3, 4, 5, 6, 7, 8. Now, if you calculate 8 plus 2 is 10. So it remaining 1 because this is 11. So what do we have? We have 1 electron in the what? Outermost shell. So sodium has 1 electron. That's why we have sodium plus, because it has 1 electron in the outermost what? Shell. Very important for you to know this. Okay. So that's everything we just discussed. All right. Very, very important. Now, sometimes, although all atoms of an element have the same atomic number, The rule number one, all atoms of a particular element has the same atomic number, all right? But all of them differ in what? Atomic mass. Some of them differ in atomic mass, all right? And if two atoms of an element differs in atomic mass, we call it what? Isotopes, all right? So different isotopes of an element have the same number of protons, but different number of what? Neutrons. It's as simple as that. Now, sometimes this isotope can spontaneously start breaking down, all right? Their nucleus can just spontaneously, disintegrate. That is what we call what? Radioactive what? Isotope. That's the one we usually use clinically. We use a lot of radioactive isotopes clinically, all right? So know that radioactive isotopes are, isotopes that just suddenly, spontaneously start, their nucleus start decaying. And when their nucleus start decaying, what do? They give off particles and energy. Extremely important. Now, this is an example of an isotope. Carbon. Carbon is 12, right? Yes. Carbon is the atomic number of carbon is 12. But if you look at this is carbon 12, carbon 13, carbon what? Carbon 14. How did they come into existence? All right. So this carbon 12 has 6 protons, 6 neutrons, making it what? 6 protons, 6 neutrons. So what is the atomic mass? 12. But this one has 6 protons. How many neutrons? 7. If you add it together, it makes it what? 13. Carbon 13. That's a different isotope of carbon. Why this one has 6 protons and how many neutrons? 8. Making it carbon what? 14. Atomic mass number 14. So this is the same carbon, you understand, the same element, but different isotopes, different atoms. So this one is mass number 12, this is mass number 13, this is mass number 14. This is what we call what? Isotope. Extremely important that you know this. This is what we call what? Isotope. Okay, so I'm not discussing these ones because this is just a medical laboratory. We use it to discover the normal person, Alzheimer's patient, CT tomography. So this is exactly what I was talking about, radioactivity, all right? Half-life, biological half-life, and all those things. Look it up later if you have time. Okay. So this is a summary. Molecule, compound, molecular formula, and structural formula, isomer. Good. Isomers is something else that they pay attention to. Isomerism. What is isomers? Isomers are molecules that have identical molecular formula but different arrangements. When we are talking about molecular formula, what are we talking about? We are talking about constitution of elements. That's molecular formula. For example, Nchebe has one mouth, 2 nose, 2 ears, all right, 2 eyes. That kind of thing. That's what is constituting Nchebe. That is what molecular formula. But my location, I am in Staten Island. That is what? Structural formula. All right, molecular formula. What constitutes me? Structural formula. Where am I? In Staten Island. That's structural formula. So isomers are molecules that have the same molecular formula. You will see, maybe I have a twin brother or twin sister or something. We have the same molecular configuration, the same eyes, the same nose, the same ear, the same mouth, the same everything. All right, but the person is in California and I am in New York. Do you understand? Or maybe the person is in Nigeria. You get the point now. So that's what we call what? Isomer. You cannot forget this. Okay. So this is an example. Look at ethanol, look at ethyl ether. All right. Look at the molecular formula for the two of them. C2H6O. C2H6O. Yes. Okay. Well, put them in the arrangement, in their location. What do you see? This is the structural formula. Different. This is alcohol. Alcohol always has OH at the end. This is alcohol. You understand? Eth, because it's 2 carbons. Then this is eth also because it's 2 carbons, but it's ethyl. You understand? You see? Because this O is in the middle. All right? So this is the difference. So this is 2 different structures, but the same molecular formula. Ethanol and ethyl ether are what? Isomers. Extremely important that you know this. Now, let's talk about bond. Sorry, I am flying. I have to because we have a lot of things to do. Now, chemical bonds. Different types of chemical bonds: covalent, hydrogen, ionic, van der Waals forces. All right, ionic attraction between anions and cations. Like if you put sodium chloride inside water now, right? Easily broken by water. Each of them are donating to each other. You understand? That's ionic bond. So when two atoms with incomplete outer shells react, each atom will what? Either share, donate, or receive. So all these bonds, either they are sharing, they are donating, or they are receiving electron. All right, that's what we call what? Chemical bonding. So the interaction, they usually results in closeness, bonding. They come close. You understand? You get married to somebody, you have your reason. Another person get married, he has his own reason. Either the two of them are sharing wealth or one is giving to the other. Do you understand? That is what we call what? Bonding, chemical bonding. It's really important that you guys understand this. All right. Now, when you're talking about covalent, what are we talking about? In covalent, the two atoms, each of them, they share. This time around, they are not donating, but they are what? They are sharing. Let me give you an example. Look at this diagram. This is hydrogen. Hydrogen H2, right? This one hydrogen, this is another hydrogen. This one has one atomos. This one has one atomos. This one refuses to give to this guy. This guy refuses to give to this guy. So instead of that, what are they doing? They are sharing. Look at it, say, okay, you know what? Let's just share. When we join heads together, we can accomplish something better. That is what we call what? Covalent bond. Extremely important that you understand this. Look at oxygen, for example. How many does oxygen have in the atomos shell? 1, 2, 3, 4, 5, 6. He has 6 in the atomos shell. This other oxygen has 1, 2, 3, 4, 5, 6. Because oxygen is atomic number of oxygen is 8, right? So 1, 2, 3, 4, 5, 6, 7, 8. This one 8 also. But this is the 2 inside. But the atomos shell is 6. This one is 6. If they are 6, I have 6, you have 6. I can't give you, you can't give me. So what do we do? Okay, you know what? Let's just donate 2 and share. Simple. That's what they do. They did rather. This one brought his 2, this one brought 2, and they are sharing and they are good. Everybody's happy. Double bond. This is single bond. This is single bond here, but this is double bond. Everybody's happy. We're good to go. All right. So that's covalent bond. We also have double covalent. All right. Like this is carbon, this is oxygen. We have CO2 that you breathe out. All right. This is how it works. Oxygen has how many? 6 in the atomos shell. 1, 2, 3, 4, 5, 6. Carbon has how many in the atomos shell? Carbon has how many in the atomos shell? 2, 4. Because carbon is 6, right? So 2 inside, then 4 outside. So carbon said, well, you know what? You, my friend, oxygen, come. You have 6, right? Bring 2 from your 6. Let me take 2 and let's share. So carbon is sharing 2 with this guy. That's double bond. Sharing 2 with this guy. That's double bond. So this one brought 2. Carbon brought 2, they are sharing. This one brought 2, carbon brought 2, they are sharing. This is what we call double covalent because there's a covalent bond here and there's a covalent bond here. Extremely important that we know this. All right? Okay. So that's by the way. I just wanted to make you guys understand. Okay, now there's something we call what? Polar covalent and non-polar covalent. Polar covalent and non-polar covalent. I'm going to share more light on this as we proceed. All right? Because you guys need to be able to what? Identify the difference. So, okay, look at what I was talking about. So this is the slide I was talking about. When you're talking about polar covalent, you are talking about what? They are sharing unequally. When you talk about, non-polar, they are sharing what? Equally. Polar, unequal sharing. Non-polar, equal sharing. Very strong bond. Carbon, polar. Hydrogen, polar. This one, polar. Equal sharing. But when you see unequal sharing, that is what? Polar bond. So I need you guys to be able to remember this: unequal, polar, equal, non-polar, and you move. All right. We have hydrogen bond as well. Water is a polar covalent bond because they are sharing unequal distribution. Now, there's something I need you guys to know: the difference between hydrogen bond and covalent bond. Let's use water for an example. All right. Water has 2 bonds. This is water. One hydrogen of water. You know, water molecule is H2O. This one H, another H, this O. One molecule of hydrogen, hydrogen in a molecule of water is bonding with another oxygen of another molecule of water. Remember that. So there are two bonds here. When one molecule is bonding with another molecule, that's one hydrogen molecule and one is bonding with oxygen molecule and another one type of bond. Now, when a hydrogen molecule is bonding with an oxygen of its own molecule, like within it, it's a different bond altogether. So if you look at these guys, you see these guys? This oxygen is bonding with in this particular molecule of water is bonding with hydrogen in this particular molecule of water. That's what we call covalent. Look at it, covalent bond. But look at this hydrogen bonding with oxygen of another molecule. That's what we call hydrogen bond. They will ask you this in the exam. You must be able to differentiate it. One molecule, another molecule, another molecule. This molecule with this molecule, hydrogen bond. But within this molecule, covalent bond. Chicken, huh? I know you're waiting for me to say chicken, huh? Okay. Then ionic bonding. You understand? This is ionic bonding. When an ion is an atom or a molecule with an electrical charge, right? When an electron is lost, a positive charge results. So ionic bonding is more like donation. You are donating. Okay, you know what? Take. I have lost. I've lost an electron. And when you lose an electron, what is it? You become positive. Remember, electron is negative charge. Remember that. So if, for example, you have a student loan and you marry somebody and you take that student loan and give to that person, what will happen to your account? Your account will become positive because student loan is what? Negative on your account. It's as simple as that. Do you understand? So that's ionic bonding. So whenever an ion, right, gives out its own an electron, what happens? It becomes positive. And when the other one, you married the person and you gave the person your student loan. So that person that took the student loan from his spouse, it will impact his account. If you have student loan of $10,000, right? I know it's not that up to that, right? I don't know. I don't think it's up to that, but as much as $10,000, for example, that person that is inheriting that student loan from you because it's married to you, the person's account is already negative $10,000. While your own is already positive $10,000. It's as simple as that. So the person that is giving out is being what? Positive. That's electron is lost. And the person that is gaining is what? Electron gain is what? Negative charge. You must understand this concept. It's a concept. All right. So when you are studying, I need you guys to play with things. Like joke with things. Just the way I'm doing. What happens here? This is chlorine. How many does chlorine have in the atom shell? 1, 2, 3, 4, 5, 6, 7. So chlorine has 7. Interesting. Now, how many does sodium have? How many does sodium have? Sodium have 1 in its atom shell. And remember, for it to get to an octet level, it has to be 8. So chlorine needs 1 to make it 8. But sodium has 1. So it's easy to throw away that 1 than to throw away 7. So what does sodium and chlorine do? Sodium says, oh, yeah, take chlorine. Take 1 to complete your atom shell. So you see, this guy, sodium, gives out this 1 to what? Chlorine. Making 8 for chlorine. So sodium becomes positive, chlorine becomes what? Negative. But the two of them together becomes complete, one molecule, which is what? They bonded together. This is what we call what? Ionic bonding, donation, covalent bonding, sharing. They can be sharing equally or unequally. As far as they are sharing, that is it. But this is what? Donation. Okay. I don't want to stress on this. So I'm jumping every other thing that talks about it. So let me see. Jumping every other thing. This is Van der Waal for us. It's a very, very weak book for us. Okay. Another thing you need to know is mixtures, because you see a question on it. All right. This is the configuration of water, for example. Water is a polar covalent bond, right? So because it's a polar covalent bond, it has solvency, cohesion, adhesion, chemical reactivity, and thermal stability. You need to know what these things are. Solvency is dissolving. It has hydrophilic head and what? A hydrophobic part of water, all right? Hydrophobic is the one that doesn't like water at all. It doesn't like water. But hydrophilic loves water, all right? So all these things are solvency. That's why it's able to dissolve fat, still dissolves sugar. Extremely important. Any party you want, it's water will give it to you. That's why we call it what? Universal what? Solvent. Now we have adhesion. You must know the difference between adhesion and cohesion. Adhesion is tendency of substances to cling to one another. Oh, I love my brother. I hold my brother very well. That's adhesion. Oh, I decide to hold the wall. That is what? Sorry, adhesion is when you are holding the wall, rather. While cohesion is when you are holding what? Each other. All right? So the bond between me and my sister is what? Cohesion. But when I'm bonding to this wall, that is what? Adhesion. I'm adhering to something. But cohesion is with each other. So, and that cohesion is what causes surface tension. When you jump up, pew, and jump inside water, bim, all right? You have to break the surface tension for you to be able to dive into it, all right? So that's why you see people putting hand like this. Why? Because they want to pierce that surface tension. All right. So that's by the way. So this is it. This is adhesion to something, but cohesion between each other. Let's not go too far into that. All right. What else do we have here? Okay, yes, we have thermal stability. All right. It's talking about being stable even with temperature. We have heat capacity. And heat capacity, this thing is something you must know. Heat capacity is the amount of heat required to raise the temperature of 1 gram of, let me see. Okay, so 3 minutes. To raise the temperature of 1 gram of a substance by 1 degree centigrade. If you want to know my heat capacity, what do? You can come and cut off my ear. Probably my ear will be weighing 1 gram, right? Then you now heat up my ear from 0 degree to 1. You understand? 0 degree to 1. That heat that took 1 gram of my ear from 0 degree to 1 is what we call heat capacity. You must know heat capacity. Extremely important. Now, let's talk about something interesting. That is what? Different substances. All right? Different substances. Look at it. We have solution, we have colloid, we have suspension. You must know the difference between these three things because they will give you a question to test you on it. All right? Now, solution consists of particles called solutes mixed with what? More abundant substance called what? Solvent. Usually water, universal solvent. All right? Solutes can be gas, liquid, or solid. But the most important thing you need to know for these three things is what differentiates each and every one of them. Look at solution, for example. Solution is under 1 nm, right? That is very tiny. Because it's very tiny, it's able to scatter light. It's not able to scatter light. Light can pass through it comfortably, no problem. So if I put a solution like this, if I pass light through it, it will pass normally, undiverted. All right? So that's solution. And also, most substances can pass through most membranes. It can pass through most membranes. And if you put it on standing, it will not separate. That's solution. Mind you, this is what? Solution. Now, compare it with colloid. When you compare it with colloid, what do you have? Colloid, a lot of proteins mixed with water, you get colloid, right? Now, colloids, their particles ranges from 1 to 100 nm. Now, look at solution under 1. Colloid, 1 to 100. Solution, can it scatter light? No. Colloid, can it scatter light? Yes. Scatter light because they are usually what? Cloudy. Particles are too large to pass semi-permeable membranes. Solution can do what? Look at it. Solution can pass through what? Membranes. Very, very important. All right. Now, what similarity do they have? Solution will not separate on standing. Colloid particles remain permanently mixed with the solvent when mixture stands, so it doesn't separate. So these three are differences between colloid and solution, but only this one, they are similar. Compare them with suspension. Suspension. When something like blood, when things are suspended in the plasma, for example, all right? Yes, like adhesive tape. Thank you so much. I appreciate. Now, these are the things about suspension. For suspension, the particles exceed 1000 nm or 100 nm rather. Solution less than 1. Colloid between 1 to 100. Above 100, suspension. Check it out. If you see this thing, you identify and you move. Don't waste your time. All right? Suspension is also cloudy. It's also cloudy. Means that, it will divert light. You understand? And it's because it's large, it cannot pass through semi-permeable membrane. The same thing with what? Colloid. But the difference between this and others is that when it stands, it separates. Like, for example, if you centrifuge blood, now for some of you that are in the lab, if you centrifuge blood, what do you see? You see the different the different substances in blood separating. That's what we call suspension. All right. And when you mix this suspension inside another liquid, that's what we call what? Emulsion. Like breast milk, fat in breast milk. That's what we call emulsion. So you need to know the differences between solution, colloid, suspension, and what? Emulsion. Now, this is acid base. You definitely have to get this. All right. The answer to this: pH of 7 is neutral. Less than 7 is acidic. Greater than 7 is what? Basic. This one is something that all of you need to know. And pH is a change of negative, a change of one number on the pH scale. Rather, pH is the measure of the molarity of hydrogen or negative logarithmic value of a particular number. All right. But they won't be asking you about pH. Instead, what they will be asking you about is substances. They'll tell you that this substances is 4 degrees. Is 4.0? Is it acidic or basic? Those are the kind of question they will ask you. All right. It's very straightforward question. Why do I have this? Okay. Now, these are different food substances with their different pH. You don't need to memorize that. Honestly, you don't. They will give you the values and you'll be able to place them. All right. Molarity. You need to know the difference between molarity and molality. When you are talking about molarity, you are talking about volume. Molality, you are talking about weight. Extremely important. This is molarity. You are talking about volume. That's why we have it in liters. All right. Then okay, that works. Then we have energy. That's capacity to do work. We have potential. All right. We have kinetic. The difference is potential is the one that is not, you are not using to do anything. It's stored. You understand? You are not doing work with it. All right? It can be chemical. It can be free. Then we have the kinetic. The kinetic is the one you are using to do work. The one you are using to move. As I'm flinging my hand now, talking, dramatizing, right? That is what kinetic energy. Too much energy is worrying me. All right? Now, example of kinetic energy, heat. All right? Okay. Then you need to know reactions, chemical reactions. These are chemical reactions. We have decomposition, we have synthesis, we have exchange. Decomposition, breaking down, synthesis, building up, exchange. They are just exchanging, sharing between each other. This is everything you need to know about chemical reactions. This is decomposition, this is synthesis, and this is what exchange. They will give you these things and tell you which one is this. All right? So this is the composition. I'm not going into details with all this. Then we have reversible and irreversible. Reversible can go in either direction. It can go this direction or it can go this direction. H2CO3, for example, can break down, to form CO2 plus water. Then this CO2 plus water can join together to form H2CO2. It depends on what it needs at that particular time. That's why we have law of mass action, which states that the direction of reaction is determined by relative abundance of substances on the either side of the equation. If I need a lot of CO2, that means CO2 is depleted. What happens? H2CO3 starts forming CO2. You understand? But if there are too many CO2, what happens? It will react with water to form H2CO3, and from there it breaks from HCO3 minus plus water and go out through the kidney. Very, very important. All these things will happen until it reaches equilibrium. Equilibrium is where the products and the reactants is stable. They are no longer forming each other. All right? And what increases these things is what we call catalyst. Increases the rate of reaction. Other things that can increase the rate of reaction is temperature, which can serve as a catalyst. All right? Okay. Then we have metabolism. Metabolism is divided into catabolism and anabolism. Catabolism is breaking down. Anabolism is building up. We have oxidation and we have reduction. Oxidation and reduction. Oxidation is in a situation whereby a molecule gives up electron and releases energy. So when I decide to give my student debt, if I give student debt, you understand, to someone, all right, so I'm giving out electron. All right, so what is it called? It's called oxidation reaction. I am oxidized in the process. I am oxidized in the process. And you that is receiving my student debt, you understand, you are oxidizing agents. You received that electron. You are an oxidizing agent. You are the person that made me lose my problem. All right? Now, reduction is the person that gains. Remember I told you, when you get negative, you are reduced. Your finance is reduced. That is reduction reaction. So if you see that this reaction, this reaction, this side of reaction is the person is gaining electron. It is reduction. And me that is donating, giving you that electron, I am now the reducing agent. I really want to be a reducing agent. I want to donate. I want to give out. I'm looking for an oxidizing agent. If any of you want to be oxidizing agent, just tell me. Let me give you the loan I have right now. Okay. So there are sometimes when these two is going at the same time. That's what we call oxido reduction. All right, oxidation reduction reaction or redox reaction. Okay. So let's talk about organic compounds. When we say a compound is organic, some people will go to the market. They want to buy meat. Oh, this meat, they need organic. Organic means that it contains carbon. Chicken, simple. If you see it in the exam, pick it. It contains carbon. And what are the organic molecules? We have the carbohydrates, we have the lipids, we have the protein, we have the nucleic acids. All right? Carbohydrates, lipids, proteins, nucleic acid. Why I'm taking time to explain this thing is because I know that most of you, it has been long you studied this thing. If I get to the chapters you did recently, I'm not going to explain like this. I'll just throw questions, all right? So I'm just giving a recap for everyone to be able to be here, all right? And if you've not subscribed, you need to do what? Subscribe ASAP. Go and click subscribe and click the notification button so that anytime I am on live or I release a new video, which I'm releasing to this night or tomorrow, you'll be able to what? See it on time. All right. So that is these are the 4 categories of carbon compounds, and carbon compound contains what? Carbon. Now let's go to these carbon compounds. It's very, very important that you know carbon compounds, which are organic compounds. Organic compounds. So you don't have to know the difference between organic and inorganic. I'm not going to explain it, but just know that organic compounds contain what? Carbon. Inorganic compounds, they usually don't even have carbon. All right, they can either be positive or negative. This one does not even have anything to do with positivity or negativity. So because of that, most of them are just sharing. So they always have covalent bond. So they are sharing. But this guy, because he's dealing with elements and all those things, that's inorganic. They are always what? Ionic bonding. All right? They are always ionic bonding. And they contain small amount of atom. Why these organic compounds contain large amount of atom? Also associated with human beings or living things. These inorganic associated with non-living things. These are comparisons which you need to go and study at your leisure. All right? These are some organic compounds. You see, they contain carbon, methane, ethane, propane, butane, and all of them are N because they have one single bond, one bond rather, that's single bond. All right. If they have double bond in, if they have triple bond iron, all right. Okay, that makes sense. So let's go into the main thing proper. All right. Now, a carbon compound can be either straight, branched, or arranged in rings. Look at it. It's straight. You see, alkane. This is alkene. You see, it's also straight. Look at it. It's straight, straight, straight. So this is straight. This is okay. This is more like branching. Then this is ring form. I'll show you more branching forms. This is ring ahead. This is branch form. Exactly. This is what I wanted to show you. This is straight. This is branch. So these are straight. This is ring form. Then this is branched. All right. This branch isopentane. This is normal pentane. When I take this C here, it becomes branched. That's isopentane. This is a closed ring pentane. That's cyclopentane. Okay. Okay, good. Remember I told you about isomers. What did I tell you about isomers? I told you that isomers are compounds that have the same molecular formula but different structural formula. So if you look at it, they have normal molecular formula but different structural formula. These are structural isomers. These are geometric isomers. These are enantiomers. Structural isomers, see, they are different. The two of them, this is butane, this is isobutane. Why? Because I removed this C and put here. But if you check it, you see that molecularly, they are the same thing. You understand? The same molecular formula, they are the same thing. All right? This is geometric isomers. Geometry means that it changed location also. All right? Look at this guy. This guy has CH3 here, CH3 in this direction. Now, this CH3 has moved here. That's what we call trans. All right? Changing from one form. If I change from this form to another form, that is transitioning, trans. All right. So the one that has in the two sides and the same direction is called what? Cis. So this CH3, I can actually put them in this part. So here now is CH3, here is CH3, then this H, this H. It is still cis2 butene. Butene, because it's in, is because it's double bond. All right. But is because it's 1, 2, 3, and 4 carbons. All right. So the same thing here. They are two the same thing, exactly the same thing, but because this one moved to one side, it is geometric. Now we have something we call enantiomers. Enantiomers is mirror image. Mirror image. Enantiomers is mirror image. Somebody said this again. Hey God, just pay attention. All right, I'm about to round up. I'm just giving a summary, just a recap. I'm making a lot of people understand this thing. So that's the idea. So we have structural isomers, we have geometric isomers, we have enantiomers. Please don't bust you're busting my bubble. So this is hexane, this is cyclohexane, this is benzene. All right. Okay. We've already discussed polar and all those things. So it's also very important for you to know this. You need to know that OH is alcohol. You need to know the carbonyl. You need to know the carboxyl groups. All right. That's acids. You need to know the amino group when you see NH2. You understand? You need to know the methyl group when you see CH3. At least this. Straightforward. So use this picture to do your wallpaper. Remove his picture from your phone or her picture from your screen and use this one. Okay. So these are just the explanation of these things, all right? Hydroxy group, hydrogen is bonded with oxygen, carbonyl, carbon linked with double bond to oxygen, carboxyl, carbon double bonded to both oxygen and what? Hydroxyl. Then this amino acid, it has nitrogen bonding. This is phosphate, it has phosphorus atom, all right? So it's just something you need to know. So these are the 4 classes of molecules that you need to also know briefly. I will just rush through it very fast and we are done. Please bear with me. All right. So when you see these molecules, we call them what? We call them macromolecules. Then we ask you, what are macromolecules? Macromolecules. Another name for macromolecules is polymers. All right. They have a lot of building blocks, strung together. All right. So they are built by what? Monomers. So you see monomer. If we are like 20 now here holding hands together, we become a polymer. But individually, we are what? Monomers. And if you see 2 of us, we are what? We are still polymer, right? Polymer is from 2 and above. Okay. So another thing you need to know is dehydration and hydrolysis. What's the difference? When you hydrolyze something, you are breaking the bond. Simple. And when you dehydrate something, it joins together. It clumps together. All right? It's just like when you are doing oatmeal. I take oatmeal so much now because of weight loss. Okay. Pay attention. Somebody is sleeping already. Please, this is just past 8 too. Don't sleep. You wait till 12 before you start sleeping. So dehydration is usually when you cook oatmeal now. If you are heating it and water is going out, it's dehydrating, it starts clumping together. Just remember that. All right. Why hydrolysis? When you pour a lot of water, you see it moving freely. All right. So hydrolysis is breaking apart. Simple. Okay. Proteins are made up of 20 different amino acids. You will see this question. It's always coming out. Protein is made up of 20 amino acids. 20. All right. So DNA is built from just 4 kinds of monomers called what? Nucleotides. So they'll tell you what are the building blocks of DNA. It is called what? Nucleotides. Simple. And you are good to go. No stress. Don't stress yourself. All right. So now let's talk about carbohydrates very fast. What do you need to know? I'm just telling you what you need to know. That's it. What you need to know, carbohydrates, they are large sugars joined together. A lot of sugars joined together. All right. Those sugars are called what? Monosaccharides. What are example of monosaccharide? We have the fructose, we have the glucose, we have the galactose. These are monosaccharides. Saccharides, sugar. All right. Monosaccharides, glucose, fructose, galactose. I mentioned it initially. Are monosaccharides. You need to know example of monosaccharides, the 3 of them. You need to know 3 disaccharides. Di, double sugar. If you look at this diagram, you see the double sugar, right? You have the maltose, you have the sucrose, you have the lactose. They will tell you one of the following is disaccharide, one of the following is what? Monosaccharide. Then these are polysaccharide. The example of polysaccharide is what? Starch, glycogen. You understand? And others. Okay. So no sweating in this. So disaccharide. I'm rushing this. These are polysaccharides. Starch is a polysaccharide. What is starch made up of? Starch. Okay, sorry. I forgot to mention something. Another thing you need to know is what makes up this what? These disaccharides. You need to know it because it's something they always ask. Where is that? This disaccharide, what makes it up? All of them are made up of glucose, so you already know one. All right. Maltose is what? Maltose is what? Somebody should tell me. Glucose and what makes up maltose? Come on, guys. Come on. Glucose and what? Are they sleeping? Wake up. Talk to me, people. What makes up maltose? It's a disaccharide. There are 2 monosaccharides that makes up maltose. What happened? You don't know it. Glucose what? Glucose. Okay. I thought somebody would tell me glucose and something else. Okay. So you need to know what makes up maltose. Sucrose is glucose, fructose. Lactose is glucose or galactose. Simple. I don't want to waste time on it. I just wanted to be sure that somebody will respond. Or you people have left me and run away. Okay. So this is starch. Starch. What's the difference between starch and glycogen? The 2 of them are the same thing. You understand? Glucose monomer. The two of them are the same thing. The only difference is just that starch is in plants and glycogen is what? In animals. So you store your carbohydrate as what? Your glucose as what? Glycogen, right? When you eat a lot, your food breaks down, carbohydrate breaks down to glucose, a lot of glucose in your bloodstream, then insulin is produced from pancreas. Insulin will convert glucose to what? Glycogen, all right? Stores it in the liver, stores it in the muscles, right? And when you need it, glucagon will break it back from glycogen to what? From glycogen back to what? To glucose, all right? And your body will use it. Then we have cellulose, all right? Cellulose is also plant. We have chitin. Chitin is usually the exoskeleton of insects. Okay, that's about it. So, second to the last. Oh, this is what I was asking. Yes, Benedict's reagents. You understand? This was what I was asking initially. This is Benedict's reagents, all right? We use it to test for disaccharides, all right? Monosaccharides, differentiate, right? Then we have tests for starch, that's polysaccharides using iodine. When it turns to bluish black, we are talking about what? We're talking about polysaccharide. Okay. I don't want to talk much about it. Lipid, what do you need to know about lipid? You need to know the types of lipid. We have fats, phospholipid, and steroids. What is the differences? You need to know the differences. Yes, glucose plus glucose, yes. What's the difference? Am I lagging? Because I said this in a long time, and you're responding now. I hope I'm not lagging. Oh. Okay. Glitz. Okay. So, you need to know the difference between fats, phospholipids, and steroids. You need to know the difference. Guys, you must know the difference. What is fat? Fat is a large lipid, right? Made up of molecules like what? Glycerol and what? Fatty acid. But the question is, how is it linked up? That's what differentiates them. Fatty acid linked to glycerol by dehydration. We know that dehydration, when you are cooking something, it joins together. We know that. Fatty acid, glycerol. Yeah, fatty acid, glycerol. Okay, thank you. Fatty acid, glycerol. Now, fats are contains one glycerol linked to what? 3 fatty acid. That is your exam question. That's what they usually ask you. I'll show you. I'll throw questions now. You guys, I know you are warming up for the questions. All of you are waiting for me. You say, this boy, talk fast. Sorry. There are a lot of people that are not grounded in this information, and I really need to explain it before we jump into the questions, because a lot of the questions are going to come out from this chapter, possibly. So, fat contains 1 glycerol linked to 3 fatty acids. Fat, 1 glycerol, 3 fatty acids. Because they have 3 fatty acids, we usually call them what? Triglycerides. Triglycerides. This is glycerol. This is 1, 2, 3. This guy join here, this guy join here, this guy join here, and that is fat. All right? Extremely important. These are different things that you guys consume a lot of these fats. All right? We have unsaturated, we have saturated fats, but I'm not going to go into it so much. All right? And when you talk about unsaturated, it's the one that has double bond. Double bonds. That's unsaturated. Double bonds. And because they have double bonds, all right, the double bonds cause bending in their carbon chain, right? That bending prevents them from packing together and solidifying at room temperature. So unsaturated fats, they hardly solidify at room temperature. Mind you, they have double bonds, unsaturated, double bonds. And because they have double bonds, it causes them to bend a little like this. So when they bend, right, they pack together too tight for them to what? They prevent them from packing together tightly, prevent them from packing too much, right? And because of that, even at a room temperature, they still remain liquids, prevent them from packing together and also from solidifying at room temperature. You have to bear in mind unsaturated fatty acids. And when you are talking about saturated fatty acids, you are talking about fats with maximum number of hydrogen, meaning that they don't have double bond. Check it out. No double bond, saturated. Most animal fats are saturated. Do you understand? So hydrogenated vegetable oils are unsaturated fats that have been converted to saturated fats by adding hydrogen. So if I get an oil and I want to saturate it, what do? I add hydrogen and it becomes saturated. Simple. And what do we call it? We call it hydrogenation. This hydrogenation creates what we call trans fats. And trans fats are always very, very associated with hypertension and atherosclerosis. We will see it, which is health diseases. Very, very important for you guys to know. So you need to know the difference between saturated and non-saturated. And why is this saturated? This is saturated because it has no double bond. The other one is unsaturated because it has double bond and it cannot bend. So it's always liquid at room temperature. You understand? But this one can bend. So this one can also be what? Solid at room temperature. And the hydrogenation of unsaturated, hydrogenated and it becomes saturated is known as what? Trans fats, or hydrogenation rather, which creates what? Trans fats. Trans, health risk. All right, I'm talking about trans fat too, please. Health risk. Okay. I know somebody's laughing at me right now, and it's okay. It's okay. It's okay. So these are the health risks. This is what research tells us. For each 5% increase. Thank you, Bronze. So for each 5% increase in saturated fat, there was a 17% increase in the risk of what? Heart disease. And for each 2% increase in trans fat, there is 93% increase in, you see? So saturated fat, 5% increase in saturated fat, there is what? 17% increase in heart disease. Saturated fat is actually causing heart disease, but the rate it causes heart disease compared to the one that is trans is a problem. All right, so the trans has the highest risk of health challenge, and that's what you should know. And I'm leaving that. We talk about fats. Now this is phospholipids. What is the difference between phospholipids and fats? What do we say about fats? We say that fat contains what? One fatty acid and a glycerol and 3 fatty acids. Right? Contains a glycerol and 3 fatty acids. While phospholipid contains 2 fatty acids and a glycerol. So the difference between phospholipids and fat is just the amount of what? Fatty acids. Chicken, they will ask you, this has 2 fatty acids attached to a glycerol. You choose phospholipids and you're good to go. Simple. And one thing about phospholipids is that because it has it has what? A hydrophilic head and a hydrophobic tail. And that's why we see phospholipid where? Majorly in the cell membrane, plasma membrane. That's where you see phospholipids. About 75% of plasma membrane is phospholipids. All right? Extremely important for you to know this. So the cluster there, they prevent water from entering into the cell. Now, the last one is steroids. Steroids are lipids, all right, which contain carbon skeleton fused together, 4 fused rings. That's steroids. They are lipids in which the carbon skeleton contains 4 fused rings. While cholesterol is a common component in cell membrane, blah, 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 blah. Single, saturated. She's laughing. Okay. Then this is anabolic steroid that people take, synthetic variants of male hormone and blah, blah, blah. This is things that the consequences it can lead to. A lot of athletes take anabolic steroid, but I'm not talking about it now because this is pathology, more or less. Now, I think this final one, proteins. All of you know proteins. How many amino acids? 20. This is a question that keeps recurring. Don't forget. How many amino acids? 20. Are you ready for the questions? Yes. I know you are ready. I know you are ready. All right. So these are everything that protein does, blah, blah, blah. It defends receptor and a lot of it. All right. I'm not going to talk more about proteins here. If you're ready for the questions, I need you to type yes. Okay. This is amino acid. What else? This is protein. Different structures of protein. These are the different structures of protein. Yes, type yes. Keep typing yes. If I get 20 yes, I will stop. So we have a primary structure, secondary structure, tertiary structure, and quaternary structure of protein. All right. Tertiary structure is always the secondary and tertiary structure are almost the most stable. Okay. Then fine. Okay, they keep typing yes until it gets to 20. So this is the nucleic acid. All right. A nucleic acid also very important polymer. You understand? Genes consist of DNA, a type of nucleic acid, but we've already done genetics, so I'm not going to talk too much on this. The gene replication happens here, DNA replicates, then transcription happens, transcription then leads to translation. All right, you need to know the locations where this thing happens. Replication happens in the nucleus, transcription happens in the nucleus, and they come out to the kitchen, right? Remember the kitchen? That's the ribosome. And that's where translation happens. You need to know the components that make this thing happen. DNA is in charge. That is the book in the library, and these ones are the RNAs. Okay, then this is nucleotides. Keep writing yes. Let me see. Is it up to 20? Don't worry, I'm not explaining others. He said, no, it's not. So this is the nitrogenous bases. I'm not explaining. Okay, that is it. Some people are like, what the hell? Yeah, I told you it's going to be long. And I'm spending more time on this because I know most of you, it has been long you did this. So this is more like a reminder. It has been long you did this, all right? Okay. So, somebody is laughing. Fed. Since you are ready for us to jump into this thing, I'm seeing only 13. You people should make it 20. This, what is it, this thing called? Is it love or like? I don't know. Anyway, that's by the way. Let's start. I see you are all gingered. So let's start. Everybody's saying yes, 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 yes. So let's start. Chemistry of life. I'm going to give you roughly 20 questions from me. So I'm not going to, or 25 questions. Okay. Okay, so testing the microphone. Guys, let's test the microphone. Everybody's here. Everybody say yes, 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 yes. Somebody said 2 hours. We've not even gotten to 2 hours yet. We are saying 2 hours. Okay, let's go. Let's go. Number 1, testing the microphone. 1, 2, 3. I play too much, and you people are also joining me to play too much. Okay, number 1, testing the microphone. Sodium has an atomic number of 11 and an atomic mass of 23. So sodium has how many neutrons and how many protons, guys? And how many, yes, how many neutrons, how many protons? Let's go. If you don't know the answer, please write not sure. Don't copy or don't follow other people. If you don't know the answer, write not sure. You have to write something. You must write something, please. I need all of yours to be active because I know some people are almost sleeping. Okay. Okay, you guys are very correct. For people that don't know how we got this, 12 neutrons, 11 protons. If you remove atomic number is number of protons, right? So just remove it from 23 and you get 12. Yeah, not sure. Yeah, so when they give you atomic mass, remember atomic mass is number of proton and number of neutrons, right? And they give you proton. Oh, they give you protons 11, they give you atomic mass 23. Just minus 23 from what? 11 from what? 23. And you will get what? Your number of neutrons. So that's how we have 12 neutrons. And remember, atomic number is the same thing with number of protons. Atomic number is number of protons. So automatically, you know that we have what? Automatically, you know that we have 11 protons. Oh, so that's it. Proton is always the same as atomic number. Yes. But mind you, it's not the same as atomic mass. Atomic mass is when you combine proton and neutron together. Okay. That's testing the microphone. So let's see. Let's see. Let me give you real questions. Solid real questions so that I'll see. You may see a diagram that is talking about difference between hydrogen bond and covalent bond in water. All right. So I'm not able to post diagram here. So, but that's okay. Okay. Try this question, guys. Still testing the microphone. No, don't think it's. Somebody will be like, stop testing the microphone. Stop it. So metabolism is the sum of dash and dash, guys. Very fast, very fast. Metabolism is the sum of dash and dash. Don't tell me you're waiting for option, no. I'm not giving you option. Hurry up, guys. Hurry up, guys. Nobody's where is my people? Don't tell me you're sleeping. Hurry up, hurry up, hurry up. Let's go. Metabolism is the sum of dash and dash. It's supposed to be two quick answers. Metabolism is the sum of dash and dash. Okay. And since you guys are giving me very weird answers, let me give you the options. A, inhalation, exhalation. B, growth, differentiation. C, anabolism, catabolism. D, positive feedback and negative feedback. E, responsiveness and movement. Let's go, guys. Okay, yes, anabolism and catabolism. The answer is C. You guys are very correct. Okay, look at this question very fast. I have to be very fast, right? Look at this question. Amylase is a digestive enzyme that breaks starch. Yes, the answer is C. Anabolism and catabolism. And in this question, amylase is a digestive enzyme that breaks starch down into sugars. Yes, you are correct. C. Into sugars through dash reaction. Bros, are you answering this question or the last one? I think you're talking about the last one. Through which reaction? Amylase is a digestive enzyme that breaks starch down into sugar through dash reaction. We have A, hydrolysis, B, dehydration synthesis, C, and anabolic, D, endogonic. Oh, okay, yes. Okay, keep going, guys. If you don't know the answer, please put not sure so that I'll be able to explain it. Because if you don't put not sure, I believe everybody understands it and I move. I fly. I jump. I'm playing. I'm just playing. Okay, you guys are very correct. The answer is hydrolysis reaction. Yes. You know, remember I told you. Yes, remember I told you when you add water to something, it breaks it down. All right. Like when you are cooking oatmeal, just remember it with oatmeal. If you put a lot of water, you see everything moving up and down. The same thing with any food you are cooking. But when that water, you are heating it and the water is drying and drying and drying, it's getting to start clumping up. All right. Okay, look at this question. Very fast, guys. Very fast. Carbon is very versatile in forming bonds with other atoms because it has dash valence electrons. Quickly. Okay, you guys are very correct. The answer is 4. He has 4 valence electron, and because he has 4 valence electron, like when it's bonding with oxygen, yeah, you see CO2, 2 oxygen, bonds 2. All right. Okay, look at this question, guys. The vibration of an eardrum is an example of dash energy. Very fast, very fast. Keep going, guys. Keep going. The vibration of an ear. Yes, you guys are right in that question is 4. The vibration of yes, you are correct in that question is 4. The vibration of an eardrum is an example of which energy? Which energy, guys? Come on, let's go. Come on, let's go. Come on, let's go, guys. Yes, everybody. We have more than 50 people in the house. Come on, guys. I need everybody to answer. Don't sleep, oh. There is no sleep for students. Somebody will be like, why am I still a student? Hey, no, when you start making the money, try remember me in your kingdom. Hey, God, okay, guys. Okay, so that's kinetic energy. Remember, kinetic energy is energy you are using to do work. See, as I'm talking, blah, 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 blah. That's kinetic energy. I'm moving my hand. That's kinetic energy. But I have a stored energy inside me, right? I'm not using it right now. That's what we call potential energy. It has potential to become kinetic, right? So look at this question, extremely important. Look at this question. An atom with 12 electrons, 13 neutrons, and 11 protons is A or an. I'm going to give options. Yes, kinetic, you're correct. You're correct. The options: A, anion; B, cation; C, free radical; D, both an anion and a free radical; and E, both a cation and a free radical. Yes. Not sure. Okay. That's good. If you are not sure, please write not sure so that I'll be able to explain. Keep going. Keep going. Not sure. Good. Please explain. Good. I will. I will. Guys, keep going. Put down the answer. If you are not sure, write not sure. If it's an emergency, write 911. If it's an emergency, write 911. Let's know how to help. Okay. Please explain. Let me see if I can get 3 more explains. Okay. Keep going. Not sure. 1. Okay. So I will explain. Good. I will explain. The answer is anion. And how do you know if it's anion or not? Just look at the question. The question says an atom with 12 electrons, 13 neutrons, and 11 protons. Normally, what is the atomic number? Atomic number is number of protons, right? And number of protons here is what? 11. Chicken. Now, if you know that it is number of protons is 11, number of protons is supposed to be equal to number of electrons. But here now they are telling that the electron is what? 12. What does it tell you? It told you that this guy collected 1 more electron. Collected 1 more electron. An electron is what? Negative. And what did I tell you about student loan? If you collect student loan from somebody, your account will become negative. And what is negative electron? Anion. Negative atom. Anion. That's how this answer is anion. Does it make sense, guys? You get the flow, right? So once you just check the number of proton, which is number of what? Atomic number. You understand? Number of proton is atomic number. Yes. They say an atom with 12 electrons, 13 neutrons, and 11 protons is what? You check the number of protons, it's 11. And number of protons is atomic number, right? It's supposed to be equal to number of neutrons, electrons rather. And that question, they told you that number of electron is what? 12. Meaning that number of electron is greater than number of what? Proton. Meaning that this guy collected 1 electron from who knows who. So he collected student debt. Even he collected student, no, isotope is a different concept, which it can be isotope. When you're talking about isotope, you're talking about different number of neutrons, all right? So, but here, this question is very well sculptured, right? It's trying to ask you about if this is positive or negative. That's what it's trying to ask you. All right. So as I told you once more, I will explain for people that maybe I'm speaking too fast. Once more, once you see 11 protons, it's supposed to be number of protons, which is atomic number, right? Now, number of protons is supposed to be equal to number of electron. So electron is supposed to be 11. But here it's 12, meaning that you gained one electron. And when you gain one electron, you become what? Negative. That's anion. Okay. I believe you guys get it. I am so confident that all of you got it. Look at this question, guys. No, isotope wasn't an option. Yeah, it wasn't. Okay, look at this question. Nucleic acids are dash of dash. A, monomers, monosaccharides. B, monomers, ATP. C, polymers, nucleotides. D, polymers, CAMP. E, polymers, DNA. Nucleic acids are dash of dash. A, monomers, monosaccharides. B, I need to increase this light of this thing so that I'll be able to see properly. Okay. So A, monomers, monosaccharides. B, monomers, ATP. C, polymers, nucleotides. D, polymers, CAMP. And E, polymers, DNA. Come on, guys. Come on, let's go. Come on, guys. Let's go. Okay, you guys are very correct. The answer is polymers, nucleotides. Not monomers, nucleotides. The answer is polymers, nucleotides. Polymers. Nucleic acids are polymers, yes. And what are they made of? They are made of what? Nucleotides. All right? You can now start saying, oh, what are nucleotides made of? I was discussing it. You people say I should stop. Nucleotides are made up of what? A ribose sugar or deoxyribose sugar. It's just made up of a sugar, a phosphate, and what? A base. A sugar, a phosphate, and a base. That's what nucleotide is made up of. All right? Okay. That makes sense. So let me ask you this question. Look at this question. You have to give me the answer. I'm not giving you the option, sorry. Because I discussed it. I don't want to give you many questions here. So I'll soon stop giving you questions, but I need to expose you to possibilities. Proteins are dash built from dash different amino acids. A, monomers 10, B, molecules 20, C, polymers 20, D, macromolecules 40, then E, peptides 25. Proteins are dash built from dash different amino acids. A, monomers 10, B, molecules 10, C, polymers 20, D, macromolecules 40, E, peptides 25. Yes, all of you are very correct. The answer is polymers what? 20. 20 amino acids. Look at this question. A drastic conformational change in a protein in response to extreme heat or pH is called dash. A drastic conformational change in a protein in response to extreme heat or pH is called dash. A, contamination. B, some people are so fast. A, contamination. B, denaturation. C, saturation. D, sedimentation. E, deconformation. contamination, denaturation, saturation, sedimentation, conformation. Fine. You guys are eating up this question, no? You are eating it up recklessly. You are destroying these questions. Yes, the answer is B, denaturation. You are very correct. So let me see how strong you guys are in figuring out my questions. Proteins perform all of the following functions except this is A, this is B. Yes, denaturation, you are correct. Denaturation, you are correct. This is B, this is C, this is D, and this is E. So what is the answer? Protein performs all the following functions except A, catalyze metabolic reactions, B, give structural strength to cells and tissues, C, produce muscular and other forms of movement, D, regulate transport of solutes into and out of cells, and E, store hereditary information. What's the answer, guys? Look at it. Okay, keep answering. Yes, the answer is E. They don't store hereditary informations. All right. They don't, guys. Good. Yes. Because the answer said except. So the answer is E. You guys are correct. Look at these questions. Triglycerides consist of a 3-carbon compound called dash, bound to 3 dash. I am not giving options. But please, any answer you are putting, you have to put 2 answers. Guys, let's hit it up. Let's hit it up. I'm not giving any options. So talk to me, guys. Talk to me, guys. Some people want option, no? Okay. The option, pyruvate fatty acids, A. Lactate glycerols, B. Eicosanoid steroids, C. D is glycerol fatty acids. E is sterol fatty acids. Answer, glycerol fatty acids. You guys are so correct. You guys are so correct. Keep going. You need to exercise your fingers so that you don't sleep. You need to exercise your fingers so that you don't sleep. So you guys are very correct. Remember, I told you that triglycerides means 3 glycerol, right? Means glycerol rather, bound to 3 fatty acids rather. So glycerol is 1, 2, 3. It's arranged in 3 layers, bound to 3 fatty acids rather. Okay. This is a giveaway. Yes, glycerol fatty acids. This is a giveaway. What is this? Proteoglycans are composed of dash. It's supposed to be 2 things. What are the 2 things that makes up proteoglycans? Hurry, guys, hurry. We don't have much time. Proteoglycans. Someone. Did you say green tea? Please don't bust my bubble this night. Don't make me laugh out with. I need energy to teach. I don't want to use energy to laugh. Okay, come on, let's go. Let's go. You want me to give you options? Nah. Proteoglycans. Two more people. Two more persons, rather. Two more persons. Come on, let's go. Let's go. One. August. Yeah. One. One more person. Proteoglycans is what? Why did you clean your protein and fat? I've seen it. Let me say protein and carbohydrates. You're correct. The answer is protein and carbohydrates. The green tree on top of the protein is okay. That's what you're talking about. You're correct. You're extremely correct. Okay, look at this question. This question said table sugar is a disaccharide called dash and is made up of the monomers dash and dash. It's actually made up of monomers dash, but I know it's two monomers, so dash and dash. So you have to give me three answers. Come on, guys, let's go. If you want options, I'll give you options. A, maltose, glucose, and sucrose. B, sucrose, glucose, and fructose. C, lactose, glucose, and galactose. D, glycogen, glucose, and fructose. E, glucose, galactose, and fructose. Table sugar is the saccharide called dash. What is table sugar? Is it maltose? Is it sucrose? Is it lactose? Is it glycogen? Is it glucose? Now the second part says, and that thing that it said disaccharides. So remember your disaccharides. You need to remember your three disaccharides. Guys, what are the three disaccharides? Maltose, sucrose, and what? Lactose, right? Yes, those are disaccharides. So the first part of the answer must be one of them. So yes, you're very correct. The answer is B. It's sucrose, and sucrose is made up of what? Galactose, glucose, and what? Fructose. So that's the answer. Not maltose and not lactose. All right. Let me see this one. Okay, look at this question, guys. You see the importance of reviewing this thing before these questions, because it helps the people that has forgotten it to remember. Look at this question. The formula of an amino group is dash. The formula of a carboxyl group is dash. You see why I told you to use it for your wallpaper? The formula of an amino group is dash. The formula of a carboxyl group is dash. Answer. Do you want options? No. Okay, let me give you options. Maybe the options will help. This is A. B is where is OH? This is B. B is CH3, NH2. Then C is OH and SH. I don't know why this thing is writing like this, but honestly, I don't know why this thing is writing like this. Then this is D. I really don't know why it's writing like this, but I will still put it NH2 CH. Okay, so A is COH, OH, B is CH3 and NH2, C is OH and SH, D is NH2 and COH, and E is SH and H2PO4. Answer. NH2, COH, NH2, COH. Someone say help. I hope the option is helping right now, because you are the one that made me put the options. D. Keep going. Two more people. Two more persons, rather. What am I speaking this night? Oh, Lord. Am I hungry? Okay, you're welcome. Yes, so the answer is what? D. You guys are very correct. NH2 and COH. So you see why I told you that amino group always have amino nitrogen, right? NH2. Then the carboxyl group is COH. Very important. All these carboxylic acids. Very, very important that you guys know this. All right. So we are gradually, I'll just ask you very few questions and we jump. We leap to the next thing. I know it's going to be long, so this is like total review. A dash reaction breaks a dash down into monomers. A dash reaction breaks a dash down into monomers. Okay, I'll give you option. Let's so that I can make this thing faster. A, hydrolysis polymer. That's A. B, dehydration synthesis molecule. Maybe I should just post it in the group instead of just reading it, because it will not make sense to some people. It's A. This is B. C. D. Okay, let me leave it at D. So a dash reaction breaks a dash down into its monomers. Keep going, keep going, keep going, keep going, guys. Keep going. Keep going. You guys are extremely correct. The answer is A. Hydrolysis breaks down. The hydration beats up. Breaks down what? Polymers into what? Monomers. Check it out. And you're good to go. So you have to be. Oh, look at this question. No. I don't know if I'm able to get this question. No, not. Am I? Maybe. So look at this question, guys. S. Hold on. Trying to get the question. Then this second H2. C is CH4. Hold on, guys. I really need to show you this question. Okay, look at the question, guys. Trying to separate it. Which of the following is not an organic compound? Which of the following is not an organic compound? So, guys, look at this thing and give me the answer. Which one is not an organic compound? A is C16H18 N3CIS. B is Na2 HPO4 PO3 brackets H2O close bra 5. Then C is CH4, D is CH3 H7 O2 and N. It's A to D. Answer. Keep going, guys. Keep going. Are you sure it's calcium? Are we looking for calcium? That's for something to be organic, it has to have what? Carbon. Yeah, carbon. You guys correct. You're very correct. Carbon. You understand? If it has carbon, okay, that's good. Usually, carbon we always associate it with hydrogen and other elements. But if you look at that B, the B has hydrogen, but it doesn't have carbon. So the important element for something to be a carbon compound is carbon. All right, organic compound is carbon rather. So look for the one that is not carbon. All right, that doesn't have carbon. If you look at all the options, everything has carbon apart from B, and that's why B is not organic. Very fast, guys. We are almost done with this. Very fast. Look at this question. Which of these functional groups contains nitrogen? Which of these functional groups contains nitrogen? A, carboxyl group. B, methyl group. C, hydroxyl group. D, amino group. E, phosphate group. Ha! Some people are firing. Oh. Somebody is on fire. I did not even stop. Say, chaka, 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 chaka. What is happening? It seems I have to go to the, okay, no. This one is important. This one is extremely important. This one is, come on, guys. I need five more people to give me the answer there. 1, 2, keep going. Which of these functional groups contains nitrogen? Two more people. Come on. Yes, you guys are very, very correct. The answer is D. Yes, amino acid, amino group rather. Okay, look at this question, guys. Very important. Very, very fast. Look at this question. Which of the following equations depicts an exchange reaction? A. I'm putting the equations, so just be patient. A, B, C, D, E, These people are so hot though. They say, Nchebe, don't waste our time. Don't waste our time. What are you doing? Okay, two more questions. Two more questions. Is it two more questions or maybe three more questions? Yeah, you guys are very correct. The answer is C. That's a reaction that involves exchange. That's C. All right. If you look at it, A plus B, AB plus CD, you know, A decided to take C, B decided to take D. That's exchange. All right. Very, very straightforward. Pray that you get that kind of question in the exam. Pray. Look at this question, guys. Now I'm going to put the options in the box. So just relax. Let me put it. Then you tell me which particular option is correct. This is A. This is B. This is C. Somebody's like, Nchebe, stop wasting my time. This is D. and this is E. Very fast, guys. Somebody has answered even before I start putting options. So let's go. In a workout, your muscle cells produce lactate, yet you maintain a constant blood pH because dash. Metabolic acids are neutralized in the muscle cells before released into blood. A. Metabolic bases are produced at the same rate by muscle cells to neutralize the acids. That's B. The respiratory system removes excess hydrogen from the blood before the pH is lowered. That's C. The body contains chemicals called buffers that resist changes in pH. That's D. Then the endothelial cells secrete excess hydrogen to prevent decrease in pH. What's the answer? I'm reading it because there are some people that will watch this later, so that in case of if they don't see this chat, they are able to, know what I'm asking. When you do exercise, you produce lactic acid in joints. That's why you fatigue. That's why you get tired. So the question is asking you how, if you produce lactic acid, that's acid. How do you maintain acid? Yes, everybody. We have up to 50 people here. Where are all of them? They have run away. Yes, the answer is D. The body contains chemicals called buffers that resist changes in pH. All right. I'll just ask you a few more questions, and that will be it. Very few more questions. Yes, buffers resist change. Thank you so much. I know why you said that. You said that so that you'll be able to remember it. I know the scope. That's a good thing, though, honestly. Like sometimes that's why I want all of you to be attempting the answers. If you attempt the answers, even if you get it wrong, you are able to remember more if you participate. If you don't participate, you won't remember. I know some people are just holding their phone now and they are at work. Very understandable. All right. If you don't participate, you should try to join tomorrow's session as well. I'll do tomorrow's session, I think, from 8 o'clock. You understand? Tomorrow's session from 8 o'clock. Almost the same thing. So if you are good with what we have today, then you don't need to join tomorrow. But if you still have time tomorrow, you can still join to increase the awareness. It's still the same question I'm going to ask. All right. But then you should be attempting it faster. All right. So look at this question. Look at this question, guys. Control X. Oh, have I already inputted the question? What did I put? Blood contains sodium chloride. Sorry. Let me see if I can delete. Okay, let me remove it. Let me put another one. Okay. Somebody's already attempting. Hold on. I really want to put this question so that everybody will attempt it. I don't know why my stuff is messing up. Just give me a moment. Okay, so that's the question. That's the question, guys. Blood contains sodium chloride, protein, and cells. All right. The NaCl is in dash. The protein is in a dash, and the cells are in dash. Options: A, emulsion, solution, suspension. That's A. B, solvents, emulsion, colloid. C, colloid, suspension, solution. D, suspension, colloid, solution. And E, solution, colloid, suspension. Come on, guys. Can you remember the differences between the three of them? You need to be able to remember the differences between the three of them. All right. And what did I say is emulsion? Emulsion is when you put your suspension inside liquid, another liquid. All right. For example, like breast milk and fat in breast milk. That's emulsion. All right. And yeah. Let's go, guys. What's the answer? Let's go. I have just 2 more questions and we are done. Okay, you guys are extremely correct. All of you are correct. Solution, colloid suspension. You guys are so correct. Look at this question, guys. Oxygen has an atomic number of 8. When two oxygen atoms come together, they form dash bond. When they come together, they form which bond, guys? That's second to the last question, and I'm giving you the last question now. Which type of bond? If you say covalent, you have to tell me which type of covalent bond. If you say covalent, because there's polar covalent and non-polar covalent. Keep going, guys. Where is everybody? Please don't tire out on me. Don't. Keep going. So yeah, you guys are very correct. It's non-polar covalent. Look at this question. Last question. This is the last question. And I'll put the options. That's the last question. This is the options A. Then this is a B. Yes, non-polar covalent. They are very correct. Then this is C. Yes, non-polar covalent. Extremely correct. This is C. This is D. And this is E. Yes, non-polar covalent. You are very correct. So look at this question. Sodium atomic number 11 reacts with chlorine atomic number 17 to become stable. In the reaction, sodium will dash while chlorine will dash. Come on, guys, let's fire. Let's fire. Okay, that's good. That's good. So the answer is sorry. Sodium will definitely give up one electron because it's 11. It has 1 in the outermost shell. So he's not doing anything with that one. It's just give it to this guy, to chlorine. Chlorine is 17. It needs 1 to complete. You guys are good. So let's jump into the next thing. Let me see if this next one I have to. Next one is what? Okay, yeah, this one is extremely fast. I'll do this one very fast. Very, very fast. This one, don't worry, I'll go very, very, very fast in this. So you don't need to worry, guys. Hope you're not tiring out here too. Don't tire out too. It's too early. Perfect. Okay. You can stretch. Cell is very straightforward. All right. Yeah. Because I'm very sure if your questions are up to 100, then cell will have a bulk. We are doing everything this night too. It's okay. Anyway, you are tired, you can go sleep. All right. I'm doing everything this night. It's not like I will do today some and I will do some tomorrow. No, I'm doing everything this night, and tomorrow I'm still doing everything. So it's just because there are some people that are not here today and they really want to be there tomorrow. So, but it's okay. Anyway, you're tired, you can go rest. Yes, absolutely. Okay, so let me jump. All right. You can throw any question you have in the box, and I'll definitely respond. All right. Now, here we're talking about cellular form and function. Extremely important that you know that all cells are composed of, all organisms are composed of cells, right? Single cell organisms, cells are responsible for the structural and functional properties of a living organism. In short, the least component of a cell is of an organism is what? Cell. Extremely important that you know this. It's involved in how your body works, the physiology, the anatomy, the pharmacology, the pathology, everything is cell. Even the therapy you give, that's pharmacology, of course. That's cell. If your cell is having a problem, that's disease. So cell study mechanism of disease as well. And the study of cell is cytology. Extremely important. You need to know just this, all right? These guys, they did a lot. They tried, all right? And that's it. Now, cell theory is extremely important to know because they will tell you one of the following is not cell theory. All organisms are composed of cells and cell products. That's the first thing you need to know. All organisms are composed of cells and cell products, all right? And the cell is the structural and the functional unit of life. An organism's structure and functions are due to its activities of its cell. That's the same thing we've been talking about. Cells are made from pre-existing cells. They are not made from non-living things. No. All right? And cells have similarities. Cells of species have many fundamental similarities in their chemical composition and metabolic mechanisms. You need to know this theory because at least you get a question from it. All right? These are the different types of cell. They may give you diagrams and they will tell you, oh, what's the name of this? So we have the flat ones, the squamous, the irregular shapes, the polygonal, the ones that looks like star, the stellate, the ones that are like cuboidal, the ones that are tall and slim, the columnar, the ones that are round, the spheroid or ovoid, the ones that are like this, discoid, the thick ones, tapered at the end, the fusiform, and also the thread-like ones, the fibrous. So these are the diagrams. You need to know this diagram because they can put this diagram for you and tell you A, B, C, D, E, F, G, H. They say, what is G? Tell them it's spheroid. You look at it. Oh, it's round, spheroid. Look at this one, looks like star, stellate. Looks like cube, cuboidal. It's flat, squamous. It's tall, columnar. It's like discoid. It's tapered at the end. You see how it gets big at the middle and tapered at the end. Fusiform, all right? And this is fibrous, like a rope. So you need to just look at it and know it, all right? You don't need to start knowing the specific micrometers. Nah, that's very important. These are the limitations of cell, all right? Surface area, volume, and others. Things that makes it grow and things that makes it not to grow. Now, you need to know this diagram because they can give you this diagram. That's the cell itself. They can give you this diagram, all right? And they point to different structures in this diagram. You should be able to know it. Prominent structures, like the nucleolus. This is the nucleolus. Where the nucleus is. This is the nucleus, all right? Right? You see it? Then this green thing is what? The Golgi. This fiery stuff is mitochondria, all right? This rough part is a rough endoplasmic reticulum. The smooth part is the smooth endoplasmic reticulum. All right. These things are the vesicle. You see how it's bursting by exocytosis and sending things outside. It's as simple as that. So you need to know this. You need to know that inside the cell is cytoplasm, right? Containing organelles, cytoskeleton, and cytosol. Three things. Organelles. You see organelles. Here's cytoskeleton. Cytoskeleton are just at the wall, helping it to have a rigidity, all right? Then cytosol is the fluid inside. Another name for it is what? ICF, intracellular fluid. Then outside it is what? The extracellular fluid. Simple. That's what you need to know. Now, they are paying more attention on plasma. Plasma is extremely important for you to know. They are paying more attention on plasma. This is the plasma. Somebody was telling me about the green tree. Yes, this is the green tree. You have to know these things because they can put A here, put B here, put C here, put D here, and tell you to choose. Diagram is part of your exam. You understand? You have to pay attention. All right. So first of all, you need to know that this is inside the cell. This is outside the cell, extracellular. This is inside the cell, intracellular. Then this is the wall of the cell. That's the plasma membrane. All right. This is the wall. Then you see this thing that looks like a rope that moves like this. All right. I don't know if my mouse is showing in your screen. Please, it's my mouse pad. My mouse pointer, is it showing in your screen? Just type yes if it is, please. So that if it's not, I won't bother using it. Yes, okay, perfect. So you see the rope, that's the cytoskeleton there. It's a protein, cytoskeleton. Thank you, guys. Now, this is the wall. The wall is made up of what? phospholipids. I told you the majority is phospholipids, about 75% or 85% is phospholipids. All right, but there are different types of lipid. One, we have phospholipid bilayer. The second one, we have the word cholesterol. These things are the cholesterol. This is cholesterol. Extremely important. Then the third lipid is the lipid that is reacting with, that's matching itself with what? With carbohydrates, which is what? Glycolipid. So once it's matching with anything, these green things are carbohydrates. All right. This is a protein. This protein is matching with carbohydrates. You see the carbohydrate tree, the green tree growing on it. That's glycoprotein. Growing on this guy, glycolipid. Extremely important that you guys know. All right. And this guy, this is channel. You see how it's open. And this one that nothing is growing on top of it and it is crossing between these two layers. This is what the transmembrane protein. And that's all. Nothing much here. And you are good to go. Phospholipids, 98% of the plasma is lipids, all right? Why phospholipids are 75% of this 98%, all right? Has the hydrophilic head and the hydrophobic tail. Hydrophilic head and hydrophobic tail, yeah, correct. Then cholesterol and glycoprotein, we've discussed it. Cholesterol 20%, glycoprotein 5%, all right? Simple, and you are good to go. All right, that's then these are other proteins like transmembrane proteins. And yes, this part is very, very important. All right, you need to know that there are different proteins. This is the receptor, this is the enzymes, this is channels, this is the identity markers, and this is adhesion protein. This adhesion protein helps you to bind to other substances. This identity marker helps you to recognize other substances. And this identity marker, it always has what? A 3 on it, which makes it a glycoprotein. Right? This is gated ion channels. They are always gated channels. They are closed. This one is what? Open ion channels. They are always what? Open. All right. Now, this is the enzyme markers. All right. Then this is the first messenger. That's the chemical messenger, which laps on the receptor. This is the receptor, the bouncer in the wall, as I always call it. So that's what you need to know. Communication is by the receptors. They come to the bouncers and they trigger the bouncers. All right. So hormones and neurotransmitters bind on these bouncers. Like if you are in my muscle class, I told you about acetylcholine. Acetylcholine comes and bind on acetylcholine what? Receptors. Acetylcholine is a phase messenger. The receptors what? And now the ones that helps in the communication inside, leading to a conformational change. So that's what receptors does. The second messengers are the ones that are inside, usually CAMP. They are the second messengers. Then we have the enzymes, all right? The enzymes help produce the second messengers. Extremely important that you know this, all right? So these channel proteins, a problem in the channel protein leads to channelopathies. I'm not talking about pathology here. I'm talking about physiology. Cell identity markers, we already talked about cell identity markers, the ones that usually have the glycoproteins, all right, which contribute to the glycocalyx. They are usually carbohydrate tagging. Extremely important. It's an identification tag. Then we have the CAMs, that are the cell adhesion molecules, all right? They are proteins that helps to bind to other proteins. So this is the second messengers. The second messenger is usually inside. Chemical first messengers like epinephrine binds to the receptor. When it binds, it activates the receptor. The receptor activates G protein, and G protein does what? G protein transfers GTP. You understand? Transfers GTP, and from there it activates adenylyl cyclase. Adenylyl cyclase converts ATP to what? CAMP. CAMP is the second messenger. So first messenger is what? That's chemical, neurotransmitter, whatever. It's just like insulin. Insulin is the first messenger. It patches on insulin receptors. And yesterday, the other day when I was teaching muscle, I told you that if there's a problem in receptor, it leads to what? Pathology, because those things will not be recognized. All right. Yes, that's when I mentioned the diabetes type 2, which happens as a result of what? Receptors of insulin not being functional enough. All right. The same thing, myasthenia gravis, these receptors not being functional enough. Okay. I'm not going to talk about pathology, guys. So this is the diagram I was talking about. This is the first messenger. If you look at it up here, it patches on the receptor. What does this receptor do? The receptor pushes the G protein. It pushes it to the adenylyl cyclase. This G protein activates adenylyl cyclase. Adenylyl cyclase in turn activates ATP, converting it to what? CAMP. CAMP is the second messenger. This CAMP is the one that will now activate the kinases. You understand? Activate the kinases. And what does this kinases do? It does every other things. It does the metabolisms, the metabolic activities inside the cell. It's just as simple as that. All right. So I will jump. So another thing you need to know is the microvilli. All right. You need to know about microvilli. Very, very important. I'll ask you a question on it. Then these are the microvilli. All right. These are the cilia also. Cilia are usually found in the respiratory tract. All right. And cilia are found in also in uterine tubes. Very, very important for you to know. That's why if somebody has a problem with cilia, the person becomes infertile. Why? Because it's also found where in the uterine tube. It's also found for women. For men, it's found in the ductus of the testis. So if somebody has a problem with cilia, like cystic fibrosis, that is, it becomes a general problem in terms of reproduction. Most of them cannot reproduce. All right. So very, very important. So there's something I want to point out. I want to point out to you because we will answer it as a question about microvilli. Microvilli are very, very tiny. That's why they are found where in absorptive surfaces. All right. Usually the broad brush border area for digestion. Microvilli. The difference between microvilli and cilia is that cilia is bigger. Cilia is 7 to 10. Microvilli is 1 to 2. Cilia is bigger. All right. And cilia is usually found here. So you need to know where each of them is being found. Then the third one is this is cystic fibrosis I was talking about. Go read it up. I'm not discussing pathology. The third one is flagella. Flagella is the biggest. All right. Like if you see the sperm, all right, you just go look for the sperm. If you look at the sperm, you see the tail and you see the flagella. You see it. You understand? Just like antenna. Okay. Another thing you need to know is the membrane transports. You need to know membrane transports, both the passive, the active, and carrier mediated. I'm not going to discuss it, but you need to learn it. All right. You shall see membrane transport. I'll ask you a question on it anyway as we move. I'll ask you. Okay, yes. Know the difference between osmolarity. Osmolarity is volume. Why? Volume in terms of volume in terms of fluid. Osmolarity is weight. I've mentioned this thing before. I remember mentioning it before. Weight in terms of kg. Okay. Now you need to know what hypotonic solution, hypertonic solution, and isotonic solution is. All right. And I remember when I was explaining this thing to my students in class, and I told you something. I told you that when you are talking about these solutions, talk about the bigger environments, not the tiny environments. All right. Talk about when they are talking about hypertonic, and isotonic. Talk about the bigger environment. All right. So it's just like this is the bigger environment, and there is something I put inside, maybe a cell that is inside this water now. So what they are asking me when they're asking me about hyper, and iso is I should compare this external environment to that little cell I put inside. Just like the extracellular fluid. Okay, so yes, this makes sense. What they're asking you when they're telling you about hypotonic, hypertonic, and isotonic, they're asking you about extracellular fluid, usually. So for isotonic is when extracellular fluid and intracellular fluid has the same number of what concentration of in terms of volume and in terms of solutes. All right. But when you are talking about hypertonic, that you are saying that the extracellular fluid has high concentration of what solutes, not solvents. High concentration of what solute, extracellular fluid. All right. So that means if it has high concentration of solute, that's high concentration of salt. If you put a cell in it, what will happen to the cell? That salt will drag out all the water from the cell, and the cell will what? Lose water and shrivel. So that's why I said whenever they're asking you this question, think of what? ECF, not ICF. It's as simple as that. That's how you get this answer. Now, when you're talking about hypotonic solution, if the ECF, hypotonic solution is when when that environment has higher concentration of what? Water, and low concentration of what? Solutes. All right. So if you put a cell, that means you are comparing that cell to the ECF. So that cell has higher concentration of what? Solutes compared to the cell. So the cell has low concentration of solutes. That's hypotonic. So what will happen to that cell inside? The cell will start dragging water inside it. It will swell and it will burst. So if you understand it that, this question is about hypertonic, about the ECF, then you'll be good. So they're asking you, this is an ECF. This is the solution. It has high salt, that's hypertonic. It has low salt, hypotonic. All right. If I put cell in it, what will happen to that cell? Will the cell take water, or will the cell lose water, or will the cell be normal? Always remember this. I don't want to spend more time here. So this is it. Hypotonic, isotonic, and hypertonic. So if I put this guy now in a hypotonic environment, that means that environment has more water than this guy. So this guy will do what? We start dragging water inside it. Start dragging water inside it. All right? Now, and it will swell up. Now, this is hypertonic. So meaning that this environment, you see, this environment is what? Extracellular. Meaning that this extracellular has more salt than inside this cell. And what will happen to this cell? This cell will start losing water and shrink. That's what they are telling you. Isotonic, it will remain the same thing. All right? I hope I have done justice to this, and I believe you understand it. So we have symport and antipods. All right? Symport, two of them going in the same direction. That's co-transport. Antipod is opposite direction. Then we have facilitated diffusion, active diffusion, secondary transport, and this. Now we have endocytosis, types of vascular transport. We have endocytosis and exocytosis. Endocytosis, I'm importing something. I'm importing to the US, endocytosis. I'm exporting to my country, Nigeria. You understand? That's exocytosis, sending out. But bringing it here is endo. Example of endo is what? Phago and pino. So phago is what? Cell eating. Pinocytosis is what? Cell drinking. They will ask you a question on this. Remember. All right? So this is an example of vascular transport. You see, this is exocytosis. Okay. What else do you need to know? Now, you should remember the cytoskeleton. Cytoskeleton is very important. It's composed of what? Microfilaments, intermediate filament, and microtubules. Microtubules helps for movement. Intermediate is for strength and support. Why? Filaments helps for other things like actin that helps in contraction and relaxation. All right? Okay. So this is microtubules. This is organelles. Different organelles. I'm not going to discuss organelles because you know organelles. The difference between rough and smooth. Rough has ribosome to it. That's making it rough. Smooth is smooth. All right? It produces lipid. Rough produces what? Proteins. Okay. You see this is rough. See how rough it is? Then this is what? Smooth. Smooth. Because it produces lipid. Simple. Okay. Then Golgi transport is Amazon. I told you about Amazon packaging. For people that work in Amazon here, you know what I'm talking about. Packages. All right? That's Golgi. All right. And another thing you need to know is that Golgi packages some vesicles that makes it become what? Lysosomes. This lysosome helps Golgi to cut things down. It's as simple as that. Some of the things is packages. Some of the proteins become lysosome. Some migrate to the plasma membrane and fuse to it, and some enter the vesicles and they push them out of the cell. So all these things are proteins that Golgi packages. Okay. These are lysosomes. Lysosomes helps to, they are a package of enzymes that are bound by a single unit. These are their functions. It helps to break down, all right? Autophagy and autolysis. Autolysis is cell suicide. Autophagy is digestion. You need to know the difference because they will ask you. And when you are talking about peroxisome looks like lysosome, but the difference is that peroxisome uses chemicals, all right? Chemicals. You talk about catalase, you talk about this, you talk about peroxide, all those things. Those are the things that peroxisome uses. But lysosomes does not use all those things. It's either autophagy or autolysis. But this guy, you start talking about what? Hydrogen peroxide, things that helps in detoxification, pushing out drugs and all those things. Oh, yes, when I talk about Amazon, I'm talking about packaging. Amazon, they package. Like, you see the people that work on Amazon, they stand up, things will come. You understand? You see it packaging seats, and sends it out. That's exactly what Golgi does. Remember the kitchen. I told you that the kitchen is where? The kitchen is the ribosome. Where is it found? There can be free ribosomes, yes, and some are found where? In the rough endoplasmic reticulum. All right? So found in rough endoplasmic reticulum. When it finishes arranging in the kitchen, it brings out to the Golgi to package. So the Golgi packages is just as Amazon packages the bag and puts it into different places. All right? Giving this one to the warehouse, keeping this one in the warehouse, sending this one to the van to take out to the people that need it. That's exocytosis. The one in the warehouse, the lysosomes and others. All right? Okay. I believe you get it now. So you know the difference between lysosome and the peroxisome. Extremely important. I don't want to talk about the powerhouse, the mitochondria. All right? Very, very important. Sometimes in the clinic, some patients are very, very funny. right? Trust me, some patients are funny. Just because patients know that mitochondria is a powerhouse. The patient came to the hospital when I was in gynecology clinic and was telling me that, doctor, I need something to give to my husband. I want the powerhouse to be working. I was surprised. I said, what are you talking about? No, she didn't say powerhouse. She said, I want the mitochondrium to be working. I said, what are you talking about? Your husband mitochondria is working. If it's not working, he cannot even talk. He cannot move because the electron transport chain happens in the mitochondria. Do you understand? I don't know that he's talking about something that will enhance the sexual activity of the husband. But that's by the way. I don't want to talk evil today. I'm of Christ. Okay, so that is beside the point. Now you have inclusions. Inclusions are the things that are found, things that are just found lying around in the cell, right? Like foreign bodies, things like glycogen, pigments, fat droplets, even foreign bodies like viruses, bacteria. All those things are inclusions, things that are included. All right, and that is end of cytology. Are you ready for my question? You see how fast this one is? This one is extremely very fast. What am I even speaking to this? Extremely very fast. Okay. Are you ready, guys? Let me see how many people are still here. How many people are still here? If you are ready, say yes, please. Let me see how many yes I get. Come on, guys. You need to activate me. Some people are saying, "Oh, Professor, I'm tired." You that is sitting down sipping juice is tired. Why me that is talking and drinking water? I'm not even drinking juice. It's not tired. Water is good for life, man. Let me see. How many years did I get? I got only 10 years. What happened? Where is all the yeses? Where is all the yeses? What am I even speaking to? Okay. You have to know how to identify cells, all right? You have to know how to identify organelles. They may ask you these diagram questions. You have to know how to identify cells, and you have to know how to identify organs. I'll start with that, all right? So let's test the microphone. Yeah, I have a lot of yes. Thank you so much. If you've not clicked this thumb, click it, all right? Please, if you are not subscribed, try to subscribe so that I don't need to be posting link. You understand? If you are subscribed and you click your notification, anytime I put live, you will see it. Because sometimes the live may not even be for you, but it may be something that you need. All right? Who is sleeping? No, I don't think anybody's sleeping yet. It's just 10 o'clock. Nah, people are not sleeping yet. Some people, their eyes is in like... That's what some people are doing right now. And they are laughing. You know yourself. You know yourself. Okay, guys. So let's test the microphone. What are accumulated cell products? They have started exposing themselves. So, what are accumulated cell products that are never enclosed in a unit membrane called? Come on, guys. Accumulated cell products that are never enclosed in a unit membrane. What is it called? Don't tell me one option. No. Only 3 people. Is that why are you texting? Some people are texting me, sending me email. Sending me email that, am I not tired? I'm not tired. Somebody's laughing, sending me email. Telling me. Okay, guys, fine. Let's business. A organelles, B inclusions, C microtubules, D microfilaments, E centrioles. What are accumulated cell products that are never enclosed in a unit membrane called? This is testing the microphone. I know, you're very funny. What are accumulated cell products that are never enclosed? Yes, the answer is inclusions. You are correct. This is just testing the microphone. Don't think you got it. Don't think it's a big deal question. This is just testing the microphone. Testing the microphone. Let's test the microphone again. Testing the microphone. Which of the following is not considered an inclusion? A. Lysosome. B. Fat droplet. C. Glycogen granule. D. Bacterium. E. Dust particle. Which of the following is not considered an inclusion? A. Lysosome. B. Fat droplet. C. Glycogen granule. D. Bacterium. E. Dust particle. It's just testing the microphone. Come on. Only 5 people. What's happening? Yeah, my people. Which of the following organelles sorts cause? Okay, no. We are still in the other question. See me moving forward already. I thought I'm still waiting for them to finish that question, right? Which of the following is not considered an inclusion? The answer is lysosome. Yes, A. Okay, look at these questions. Testing the microphone. Look at this question. Quickly. I want us to finish this and jump. Which of the following organelle sorts, cuts, splices, and packages newly formed proteins into membrane-bound vesicles? Somebody is not sure. Is it Golgi? Is it Golgi? Yes, the answer is Golgi. You know why I like cells? Cells answers are very, very more. Most of the questions are testing the microphone questions. Look at this one. Which of the following organelle is not involved in protein synthesis? I'll give you option A, smooth ER, B, rough ER, D, C, Golgi complex, D, the nucleus, and E, ribosome. Which of the following organelle is not involved in protein synthesis? A, smooth ER, B, rough ER, C, the Golgi complex, D, the nucleus, and E, ribosome. Answer. Exactly. The answer is smooth ER. You guys are so hot this night. Is it because I'm giving you test the microphone questions? Hold on, I'll still give you very tough question now, and you beg me for the answer. Which of the following decomposes fatty acids and detoxifies alcohol, free radicals, and drugs? You have to beg me for the option. Okay. A, lysosome. B, microvilli. C, inclusions. D, peroxisomes. E, Golgi vesicles. Somebody begged. Which of the following decomposes fatty acids and detoxifies alcohol, fatty radicals, fat-free radicals, and drugs? A, lysosomes. B, microvilli. C, inclusions. D, peroxisomes, and E, Golgi apparatus. Peroxisms, extremely correct. Let's see. Okay, you guys are proving hard not to crack. That's what you guys want to show me. You are proving hard not to crack. I will crack it. Watch. You can't defeat me this night. You are just so funny. I see that. Dash. There's different types of smile. No. You don't understand. Dash synthesizes carbohydrates and puts finishing touches on protein. Dash synthesizes. Let me use my large screen. Dash synthesizes carbohydrates and puts finishing touches on proteins synthesized at Dash. So there are two answers here, guys. There are two answers here. Amand, there are two answers here. Dash synthesizes carbohydrates and puts finishing touches on protein synthesized at Dash. What is the answer, guys? There are two answers. So if you are giving me, give me two answers. I'm not giving you options, sorry. First one is the one that synthesizes carbohydrates, and the second one is the one that puts finishing touches. That's how you get the answer. So if you give me anyone you give me first, that's the one I will take in the first option. Okay, you guys are so correct. So Dash synthesizes carbohydrates and puts finishing touches on proteins synthesized at Dash. Dash synthesizes carbohydrates and puts finishing touches. So the first one is Golgi complex. Golgi complex. You understand? Because it's the one that puts finishing touches and protein synthesized at the rough ER. So the answer is the Golgi complex and rough ER. Okay? Yeah. Look at this question. Yes, Golgi and rough. Perfect. You're good. Look at this question. Which organelle synthesizes steroids in the ovary and stores calcium in muscle cells? Which organelle is that? Which organelle is that? Come on, guys. Let's hurry it up. Let's hurry it up. Some people are already getting tired. Somebody just yawned now, and I heard that yawning. I heard it. I heard it. It's me. I know. I heard it. I heard it. I was shocked. It was sounding like a volcano. Okay. Come on, guys. Come on, guys. Which one synthesizes steroids in the ovary and stores calcium? So the hungry yawn, extreme hunger. Okay, it's not tired yawn, it's hungry. Oh, hey, yeah. Just use Uber Eats. Okay, so the answer is smooth endoplasmic reticulum, all right? It's the one that produces lipid, and all these things we mentioned are lipids, all right? You see, these questions are very straightforward because of the fact that we've already conquered it. It's pretty straightforward. Look at this question, guys. Which organelle is most active in causing programmed cell death? Answer. Let's go very fast. Say lysosome. Options: rough, ER, nucleus, centrose, lysosome, Golgi. What's the answer, guys? Come on, everybody, type. Don't sleep on me. Don't doze. Exactly. You guys are so correct. The answer is lysosome. You need to note these things. All right? Now, look at this question. It's a pathophysiology question, and I need you guys to attempt it. Which function would immediately cease if the ribosomes of a cell were destroyed? Exactly. You guys are so right. It's protein synthesis. You guys are so right. Which of the following gives a cell structural support, determines the shape of a cell, and directs the movement of substances through a cell? Yes, that answer is protein synthesis. So I'm jumping to I'm trying to be a little faster, all right? Because especially muscle. I need to cover muscle with you guys this night because I've not done it with you guys. What's the answer to this one? Which of the following gives a cell structural support, determines the shape of a cell, and directs the movement of substances through a cell? Okay, this is interesting. Let's see who gets the answer. I'm seeing two answers there. I was expecting like 4 different answers. But let me see who gets it. Make sure you subscribe. Also, make sure you on the notification buttons so that when you are doing AP2, even if I'm not able to send you the link, you'll be able to see when I start my live sessions, because henceforth I'll be doing live sessions for every level. All right. So, in case of if you are not with me for AP2, so that you'll be with me on live always. Some people are wondering, if you are not sure, write not sure. Instead of not writing at all. If you are not sure, write not sure, guys. I need you guys, everybody to be active and engaged. Not sure, write not sure. Or 911. Which of the following gives the structure? The answer is what? Cytoskeleton, not plasma membrane, guys. Yeah, it's fine that you said not sure. The answer is not plasma membrane. The answer is what? Cytoskeleton. Cytoskeleton is that rope inside that helps to give it structural support and determines the shape of a cell. I mentioned it when I was discussing. Okay, let's see this question. Okay, now this is a major question. Yes, cytoskeleton. Now this is a major question for you. Let's see if you are able to get the answer. This. Okay, let's see. A red blood cell is placed in a high power tonic solution. This means the concentration of the solutes in the solution is dash than the concentration of solutes in the intracellular fluid and will cause the cell to dash. Answer A. Let me give you the option, guys. This A, higher swell. B, lower swell. C, higher shrink. D, lower shrink. C, lower bursts. Yes, you guys are so correct. The answer is higher shrink. I've explained it. I'm not, and nobody said not sure. So I believe all of you understand it. So I'm not explaining again. Look at this question. This is the question here. All right. This is A. This is B. This is C. This is D. And this is E. Answer. Two solutions are separated by a selectively permeable membrane. If solution A has a higher concentration of non-permeating solute than B, then dash. A, the solute will pass down its concentration gradient from solution A to solution B. The solute will pass down its concentration gradient from solution B to solution A. C, the water will pass down its concentration gradient from solution A to solution B. And D, water will pass down its concentration gradient from solution B to A. And E, neither the solute nor water will diffuse. Answer. Hey, only one person. What's happening here? This is challenging. It's not that one that all of you are busy answering and getting it very fast. Let's here be something that will challenge people. Come on, guys. Come on. Come on, come on, come on. Okay, so the answer is actually D. Water will pass down its concentration gradient from solution B to solution A. All right. Water will pass down its concentration gradient from solution B to A. This is just osmosis. All right. The key things you need to note in this question is the solute is non-permeating. When we say non-permeating, it cannot cross the membrane. That is solute. All right. The solution A has more solutes. That means solution A is what? Hypertonic. Right? Solution B has less solutes. It's hypotonic, means it has more water. So what will water do? Water will move down its own concentration gradients. High water will move to low water. You understand? That means from solution B to solution what? That means water will pass down from solution A to solution B. All right? So the answer is what? D. From solution B to solution A, rather. Water will pass down its concentration gradient from solution B to solution A. So it's very straightforward question. It's just osmosis. If you pay attention, you understand what I'm talking about. Look at this question. Which of is transport LDL uses to enter the cell? No. What type of solution contains higher? Look at this question. Where is everybody? You guys are feeling sleepy. When I've not even started muzzle. Better don't sleep. Oh. I've never done muzzle in life before, and I've not done the video. Have I done muzzle video? Yes, I think I have. The video I need to post this night or tomorrow is bone. So which type of solution contains higher than normal amounts of osmotically active solutes? Yes, bone. I will post either bone this night or tomorrow. I've not done the video, but I've already produced the slide for it and the questions. Hurry up. I need to ask 10 more questions and I'm done with the Excel. You understand? Just 10 more questions. Let's hurry. We need to hurry. If we waste time, if 10 people don't answer, I'm not going to move forward. So you guys need to answer. I need to be sure that all of you are here. Come on, guys. Hypertonic. Yeah. This is a giveaway question now. So this is also another giveaway question. Very fast, guys, because the faster the better. Look at this question. Thank you. Yeah, that's hypertonic. Which of these solutions, which of these processes brings droplets of extracellular fluid into a cell? Hurry. Answer. No. That thing is not hypo. It's hypertonic. Oh. Which of these processes brings? No. Not even this one. I thought you were answering the other question. Which type of solute contains higher than normal amount of osmotically active solutes? That one is hypertonic. Oh, is it this question that you're answering? Which of these processes bring droplets of extracellular fluid into a cell? Is that what you're answering? No, I don't think so. Hypotonic? Why are you people saying hypotonic? You guys are so funny. Why are you guys saying hypotonic? Everybody's saying hypotonic. Oh. Are you sure is this osmosis or you mean types of transport? The question is asking about transport. Which of these processes brings? Okay, I should give option. Let me just give option. A is phagocytosis. A is phagocytosis. B is receptor-mediated endocytosis. C is transcytosis. D is pinocytosis. E is exocytosis. Phago, receptor-mediated, transcytosis, pinocytosis, and exocytosis. Answer. Honestly, when you guys are saying hypotonic, I was like, what happened? Why did they change from hyper to hypo in the other question? I didn't even know it's the one I asked now that you guys are answering. Okay, let's go. Hurry. Yes, the answer is pinocytosis. Very correct. Very correct. So I need to just I need to move on to this other question. I'm trying to hurry it up. This question, look at this question. Which of these is an example of active transport? Yeah, which of these is an example of active transport? I will put the options. This is A. This is B. This is B. This is C. This is D. And this is E. Answer. Which of these is an example of active transport? Which of these is an example of active transport, guys? A, the diffusion of oxygen from a place of higher concentration to a place of lower concentration. B, the facilitated diffusion of potassium. C, the transport of glucose down its concentration gradient. D, the transport of sodium from a place of low concentration to a place of higher concentration. And E, the transport of chlorine from its concentration gradient. The answer is D. You guys are correct. Sodium transport, the transport of sodium from a place of low concentration to a place of higher concentration is by using ATP. Remember when I told you about sodium jumping? It will drag potassium and jump back into the blood, all right, during reabsorption. That is active transport. It uses energy to drag potassium towards it. It's a co-transport, sodium and potassium. Antiport transport, not co-transport rather. It's an antiport because it drags sodium into the space and jumps in. All right, it drops potassium rather. But mind you that sodium also transports itself with glucose, but that one is not active transport. But the one that it exchanges with potassium is the active transport because it is in ATP. So the transport of sodium from a place of lower concentration to a higher concentration is active transport. Okay. So I have very few more questions. What type of observation was this? Very few questions to ask you. Pinocytosis requires use of energy. You guys know this one. Which one requires use of energy? Active transport. Oh. Okay, look at this question. Very important. It looks like a question that somebody will love. So, what do facilitated diffusion and active transport have in common? What do they have in common? A, B, C, D, E. Answer. What do they have in common? What do they have in common, guys? Everybody. You have to type something. You have to, if you don't type something, I'm coming there now. Type something. Anything you type is fine. You have to type something, guys. Everybody. What do they have in common? Yes, you guys are very, very, very correct. Both of them are example of carrier-mediated transport. All right? You guys are extremely correct. Now look at this question. Look at this question, guys. Very fast. It has two answers. Sodium and glucose are transported together from the interstitial lumen into an interstitial cell. Intestinal lumen into the intestinal cell. The carrier protein is A dash. Sodium and glucose are transported together from the... What? No, hold on. I made a mistake in this thing. Hold on, please. Hold on. Let me remove it very fast. I made a mistake. Sorry. Look at the question here. Very fast, guys. Sodium and glucose are transported together from the intestinal lumen into an intestinal cell. The carrier protein is a dash, and the process is called dash. I'll give you options. Pay attention to the options if you want. A, symport co-transport. B, symport counter-transport. C, antiport counter-transport. D, antiport co-transport. E, uniport facilitated diffusion. What happened? Is the gas finished? Sodium and glucose are transported together from the intestinal lumen. I just mentioned it some moments ago. That's why I asked this question. I just discussed it some minutes ago. What's the answer, guys? Energy. Where is the energy? What happened? Energy. Two more people or three more people? Yes, you guys are very correct. The answer is A. I told you that sodium glucose, when they are going together. Both of them are going together. All right, that's a symport co-transport. They are going together. But when it is sodium and potassium, sodium is always deceiving potassium. It will drag potassium and it will jump. That's antiport, and that one is the one that is what active transport, because it needs energy to drag it. But sodium and glucose, they are just going together. They don't need it. It doesn't need energy to do that. So it's not an active transport. All right. So this one is symport co-transport. That's A. Extremely important that you know this. Okay. Look at this question. Guys, look at this question. Insulin is taken up via endocytosis by the endothelial cells that line the blood capillaries. Then it's transported across the cell to the other side where it is released. The transport is called dash. Option A, pinocytosis. B, receptor-mediated phagocytosis. C, passive transport. D, facilitated transport. And E, transcytosis. You ask yourself, if I am in the exam hall and they ask me this question, will I be able to get the answer? You understand? So you have to up your game. If you are blinking, open your eyes. Do like, say like this as you are reading. Now as I'm typing, just put two of your hands like this if you are sleeping. All right? So that you don't sleep. You must not sleep. Come on, guys. Make attempts. Make attempts. If you fail it, that's fine. Make attempts. Everybody has to be active, guys. I just find out that there are just select few people that are active here, and that's not right. All right. I need everybody to be active. You have to pick an answer. Don't just listen. You see, you understand? If you just listen, you will not retain. You understand? When you are selecting answers, you see yourself reading that question, analyzing it very fast, and be able to generate an answer. All right. Don't just look. When you look, oh, I think I know it. Okay, I know the answer. Oh, everybody's choosing it. You will remember. You have to put it. All right. Someone said extra points. Please, the answer is transcytosis. What point are you going to give me for being awake right by this time? Extra points. Okay, so extra points. Yes, it's a transcytosis, guys. So look at this. Look at this. Dash is the process in which dash forces water and small molecules, small solutes, such as salts, through narrow clefts between capillary cells. I'm asking you 4 more questions, and that will be it for this. Dash is the process in which dash forces water and small solutes, such as salts, through narrow clefts between capillary cells. A, filtration and hydrostatic pressure. B, osmosis and hydrostatic pressure. C, active. Let me just post it in the group. This is A. This is B. This is C. Everybody, guys. C. This is E. D. And this is E. What's the answer? Dash is the process in which dash forces water and small solutes. Yes, the answer is A, filtration and hydrostatic pressure. Filtration and hydrostatic pressure. You are correct. Cellular membranes are permeable to. You're very correct. Okay, perfect. Look at this question. Because I told you guys about this, and I really need you to know it so that if there's any question like this, you'll be able to answer. Yes, you are very correct. The answer is A. You are very, very, very correct. The answer is what? A. Look at this question, guys. Yes, the answer is A, filtration and hydrostatic pressure. The plasma membrane of cells lining the small intestine has dash, which are specialized for absorption of nutrients. Is it flagella? Is it cilia? Is it microvilli? Is it glycocalyx? Is it dianin arms? The plasma membrane of cells lining the small intestine has dash, which are specialized for absorption of nutrients. Is it flagella? Is it cilia? Is it microvilli? Is it a glycocalyx? Is it dianin arms? Answer. Come on, give me the answer very fast, guys. Quickly, quickly, quickly. I want us to be done with this. Perfect. The answer is microvilli. All right. right? The answer is microvilli. Then look at this. If you think you know, you got that answer, oh yeah, try this. There are three answers here. Three. So I need you to write the three of them in one line, in one sentence. Don't write it, do this one, this one, this one. No. Write it in one sentence. Yes, microvilli. Yeah, you're correct. You're very correct. Come on, guys. Dash provide motility to a sperm. Dash acts as sensory antenna in many cells, and dash increases cell surface area. You guys are so correct. The answer is flagella cilia microvilli. So flagella provides motility to sperms. Cilia acts as sensory antenna in many cells, and microvilli is usually found in the digestive system for absorption. Extremely important. Extremely important. Okay, you think you know it? Fine. You got it. I'm going to defeat you guys with that. What's the answer to this? The coat of carbohydrates on the outer surface of the plasma membrane. Yes, you're correct. You're correct. You're correct. Flagella cilia microvilli. The physical coat of carbohydrates at the outer surface of the plasma membrane that functions in cell identification is called dash. A, brush border. B, glycocalyx. C, cholesterol coats. D, phospholipid bilayer. And E, cell adhesion molecule. Yes, you are correct. Flagella cilia microvilli. You guys are so correct. The answer is glycocalyx. You guys are so correct. Look at this question. Look at this question. In the second messenger role, CAMP activates enzymes called dash, whose job is to regulate other enzymes by adding phosphate groups to them. If you remember what we discussed, after CAMP, what was the next thing? What is the name of those enzymes? What is the name of those enzymes? Keep going, guys. Is it just these 5 people that are awake? Are you people eating? Don't eat without me. If you want to eat, you have to invite me to that place. You can't just be eating. Yes, the answer is kinesis. You guys are extremely correct. So I want to ask you, I think, 2 questions, and that will be it. And that will be it. All right. You guys are so correct. Look at this question. The plasma membrane is mostly made of molecules that have hydrophilic heads and hydrophobic tails. These molecules are called dash. Yes, kinesis, you're correct for that other question. Keep going, keep going. This is second to the last question here. Yes, you guys are so correct. I like the activity. See, there are more than 10 people now that are answering. You people have to be encouraging me. Oh. All right. If you are not answering, if 2 people are answering, I will not be encouraged. I need encouragement. Okay, cells that tick in the middle and tapered. This is the last question, guys, for cells. Cells that are tick in the middle and tapered toward the end are called dash. Somebody said, I can't wait for this guy to finish. Are called what? Thank you so much. Okay, yes, it's called fusiform. So that will be it. All right. And I really need you guys to, when maybe like tomorrow or next or whenever, you come back and look at these questions. I know why. I know why. All right. Because for people that will be in my class, your midterm is 100 questions, and a lot of these questions are or something similar will be there. So I want us to jump. All right. Since we did genetics recently, I want us to jump to muscles. So let's come from the last things we did backwards. You understand? Because I know some people are already getting so tired. So before you get tired, let it be something we've already revised. So I want to do muscles and bone because we've not done anything on muscles and bone. All right. And I'm not going to explain it. I think I should just throw in questions. Who is with me? I should throw in questions, or you want me to just go through the slide? I don't think it's necessary because we just did it recently. Should I just go straight to the questions, right? Please, I need somebody to support me. It's not like I'm tired, though. Trust me, I'm not. I can talk to money. As far as I have the time. It's just that I don't have the time. All right. So, question. Good. Perfect. So we don't need to start. So you guys agree that we should muzzle, then go backwards, right? The last thing we did was muzzle, right? Yes. Is it not muzzle? Yes, muzzle. So I think we should do it backwards so that, because I know we've already done revision not too long ago. You understand? So for the other ones, but we've not done for muscle and bone. So I think I should just jump into muscles and hey, oh, you have work in the morning. I'm so sorry. But it is what it is, eh? Sacrifice. Me too. You say you haven't worked in the morning like I'm not working. Me too. Me too. Okay. So let's go. Testing the microphone. Michael Jordan was arguably the best player in professional basketball history. He excelled at jumping and sprinting down the court. Anatomically, one would expect him to have highly developed dash fibers in his lower limbs. Is it red? Is it type 1? Is it slow oxidative SO? Is it fast twitch? Is it smooth muscle? Answer. A, red. B, type 1. C, slow oxidative. D, fast twitch. And E, smooth muscle. What's happening? This is just testing the microphone, guys. We are testing the microphone. Hello. Say clinicals in the city. It's fine. Drink coffee. Hey, God. Drink coffee, or like you can go off and still join tomorrow evening. Tomorrow, I have a class till 9, so but I will try to finish on time my class so that I can start by 8. So tomorrow, I most probably will start by 8. Somebody say 911. Hey, this is testing the microphone. This is testing the microphone. A, red. B, type 1. C, slow. D, fast twitch. And C, smooth. You see the advantage of reviewing this thing before we start answering questions. This is the advantage. But you don't think about the advantage. Instead, you're more interested in let's just answer this question and get out of here. Microphone has broken. Oh God. Talk to me, guys. Yes, it is. Yes, it is. You are still correct. Same microphone has broken. Anyway, the answer is fast twitch. All right, it has to be very, very fast. That's white. You understand? Endurance is the red one, which is the red one is slow, oxidative, and also is type 1, but type 2 is fast. And the smooth muscle there does not make sense because what we are talking about is skeletal muscle. So that's this testing of this microphone did not go well. Oh. I don't understand. What happened to the microphone? Did it break? Yes. So look at this question. You guys have to wake up, because let me tell you why the other ones we were doing was so easy for you guys. Because like first of all, I reviewed them. I discussed them before we started. All right. So this one may be a little tough for you because I didn't review it. All right. So that's why you need to wake up right now. If you are sleeping, do like this, do like this, do like this. I should probably have started with this. Maybe. Okay, look at this question, guys. Drugs called calcium channel blockers may be used to lower blood pressure by causing arteries to vasodilate. How do you suppose these drugs work? Let me give you the options. Just pay attention. A, B, C, D, E. Yes, white and fast twitch. Yes, you're correct. That's type 2. It's fast oxidative. So drugs called calcium channel blockers may be used to lower blood pressure by causing arteries to vasodilate. How do you suppose this drug work? A, they prevent calcium from entering the sarcoplasmic reticulum of smooth muscle. B, they stimulate the calcium pump in smooth muscles, thus removing calcium from camodulin. C, they prevent calcium from exiting the sarcoplasmic reticulum in skeletal muscle. D, they prevent calcium from entering cardiac muscle, thus slowing down the heart rate. And D, they prevent calcium from entering smooth muscle, thus allowing the muscles to relax. Come on, guys. Only 4 people. This microphone has scattered. There's nothing anybody can tell me. It has broken. It has shattered. Don't tell me this muscle is more difficult for you guys, man. Yes, guys, the answer is E. They prevent calcium from entering smooth muscles, all right? Specifically, thereby slowing down the muscles, thereby allowing the muscle to relax. Everything is all about relaxation. And remember, they told you arteries, that is smooth muscle. You understand? All these ones that is telling you preventing cardiac. You understand? Well, we don't have anything to do with cardiac. You understand? We are talking about arteries, that's smooth muscle. All right. Then the C is talking about skeletal muscle. No. All right. Which one again? Okay, A and B is talking about smooth muscle, but this one is talking about entering sarcoplasmic reticulum. This one is talking about pump. No. So the answer is E. They prevent calcium from entering smooth muscle, thus allowing the muscle to relax. That's vasodilation. All right. Okay. This question. Look at this question, guys. I don't understand why this microphone is breaking this night, though. Is the question too difficult? This is the question. Which of the following is an accurate comparison of skeletal muscle to smooth muscle? These are the options. A. B. C. D. What is happening, my people? E. Okay. What's happening? So choose if it's A, B, C, or D. Which of the following is an accurate comparison of skeletal muscle to smooth muscle? A. Smooth muscles contract more slowly but relaxes more rapidly. B. Smooth muscles contract more rapidly but relaxes more slowly. C. Smooth muscles contracts and relaxes more slowly. D. Smooth muscles consumes more ATP to maintain a given level of muscle tension. And D. Smooth muscle depends. E. Smooth muscle depends on an axon to supply the calcium needed for contraction. Only two people. What is happening? Okay, you guys are very, very correct. The answer is C. Smooth muscle contracts and relaxes slowly. It's a simple thing. It's actually very straightforward because skeletal muscle every time is moving very fast. Everything fast. But everything about smooth muscle is what? Slow. It's gentle. You know, you see, smooth muscle, very, very gentle. Like me, I'm very gentle. I don't care what you say. I know you're laughing. That's fine. Okay, look at this question. Which muscles can contract without the need for nervous stimulation? A. Skeletal muscle only. B. Smooth muscle only. C. Cardiac muscle only. D. Smooth and cardiac muscle. E. Skeletal, smooth, and cardiac muscle. Yes, you guys are right. That one is C. Smooth. Okay. Did you say what? Bronze. I don't know what to say. So I want you to choose A, B, C, D, or E. A. Skeletal muscle only. B. Smooth muscle only. C. Cardiac muscle only. D. Smooth and cardiac muscle. E. Skeletal, smooth, and cardiac muscle. Okay, so see, some people are you choose smooth. Is your smooth muscle only, or smooth and cardiac muscle? Like they are smooth in the two of them. They are smooth in the three of themselves. Even skeletal, smooth, and cardiac. E. So I need you to choose A, B, C, or D, or E. Skeletal muscle. I'll repeat again. A. Skeletal muscle. B. Smooth muscle. C. Cardiac muscle. D. Smooth and cardiac muscle. E. Skeletal, smooth, and cardiac muscle. Answer. Come on, guys. Let's fire. Let's fire. Let's fire. Yes, the answer is D. The answer is D. It's smooth and cardiac muscle. Which muscles can contract without the need for nervous stimulation? The heart does not need nervous stimulation. It just contracts and relaxes. The same thing with smooth muscle. The one that needs nervous stimulation is your skeletal muscle. That's why if I touch you, the nerve will give information, pass, come back. You will now move. You will now contract. All right? So please, you need to understand that question properly. It's very, very important. Okay. Partly to increase, no. Okay, what's the answer to this? Very fast, guys. Don't worry, it's getting easier. The microphone has broken. Come on, guys. What's happening to all my people? Resistance exercise. What's the... I want to point like very fast 5 questions now, but you need to move with my pace so that we can get this thing done quickly. Yes, you guys are so right. That answer is hypertrophy. Yes, I'm not going to discuss it. That answer is hypertrophy. Look at this question, guys. Loss of muscle mass from lack of activity is called dash. Quickly. Okay. Now people are gingering. I love it. Quickly. Yes, you guys, loss of muscle mass from inactivity is what? Atrophy. You guys are so correct. Look at this question. Yes, you're correct. Which of the following has the largest mitochondria? Which of the following has the largest mitochondria? Yeah, Sophia, you're right. Which of the following has the largest mitochondria? We have A, slow oxidative fibers, B, fast glycolytic fibers, C, cardiac muscle, D, smooth muscle, E, intermediate fibers. Which of the following has the largest mitochondria? A, slow oxidative, B, fast glycolytic, C, cardiac, D, smooth, and E, intermediate. Quickly. Me? You don't know me. I'm naturally hyper. I don't need any external thing to make me hyper. I'm naturally there. Funny enough, I can be here till the next 2 to 3 hours, and I will still wake up first in the morning. Terrible. Yes, the answer is cardiac. Absolutely, the answer is cardiac. All right, because it needs to keep pumping. You understand? So you need energy. So it has the highest mitochondria. This one is a given. All right. This one is a given. Look at this question, guys. I'm just going to give you guys 3 options. Just 3 options. I don't want to be putting in so many options here. This is A. This is B. No, let me put all the options. This is B. Yeah. This is C. This is D. And this is E. Why do you continue to breathe fast after rigorous physical activity has stopped? A. The diaphragm is waiting for its acetylcholine to run out. Terrible. But okay. B. The body requires more oxygen to restore levels of ATP and creatine phosphate. C. The lungs can't stop breathing until the heart rate slows down. D. The myosin heads need more oxygen to fully detach from actin. And E. The kidneys require oxygen to dispose of excess lactate. Yes, you guys are so correct. The answer is what? B. The body requires more oxygen to restore levels of ATP and creatine phosphate. If you remember when I discussed creatine, if you don't know a lot of things in muscle, please go and watch the muscle video. There's a video there for muscle. Yes, you are correct. The answer is B. Look at this question. Which of the following systems would provide? This one is a given. Which of the following system will provide energy for a racquetball player while playing a point? A. The respiratory system. B. The glycolytic system. C. The phosphagen system. D. The anaerobic system. And E. The glycogen lactate system. Answer. When playing a point, so burst of energy. Is it the respiratory system? Is it the glycolytic system? Is it the phosphagen system? Is it the anaerobic system? Is it the glycogen lactate system? Come on, guys. Come on. I need us to answer faster because we have things to do. Come on now. Just one person? What's happening? Am I in the wrong class? This is the first time I ask question and only one person answer for a very long time. What is happening? We are still testing the microphone. I don't know why you are not answering. Repeat options. I can't even remember the options I called because I've left that question. I'm in another question I need to ask. I honestly will not be able to repeat it. Sorry. But yes, the answer is phosphagen. Right? For the person that answered phosphagen system. You remember phosphagen system? You should be able to remember phosphagen system. All right? That's how the creatine phosphate, collects the kinase is collected, creatine kinase rather. CP. You remember CP if you can remember very well, where the kinase collects phosphate from one ATP, leaving it as what? From one ADP rather, leaving it as AMP and giving to another ADP. Do you understand? Which makes it what? ATP. So you have that burst of energy. That's what they are just asking you. It's phosphagen system. There's nothing like respiratory system to even start with in terms of that. All right. It's phosphagen system. Please look at this question very fast. All muscle cells will respond to an electrical stimulus because all muscle cells are dash. A. Excitable. B. Contractile. C. Striated. D. Involuntary. And E. Isometric. I'll be reading the options just once because I have to move to the next question to make this thing faster. All right. Answer. All muscle types will respond to an electric stimulus because all muscle types are what? Excitable, contractile, striated, involuntary, isometric. Answer. Come on, guys. This one is easier now. I need everybody to be answering. And not just to be answering faster. Yes, it's excitable. Look at this answer question. Come on, guys. Look at this question. A bundle of muscle fibers is known as dash. Come on, guys. Come on, what is it? Now activity has started. I think the microphone is back. Hey, Jehovah. I think the microphone is back, right? Okay, so let me give you guys more microphone questions because, like, why do I need to? I don't have to stress you, man. Look at this question. The sarcolemma of a resting muscle fiber is most permeable to what? A, calcium, B, potassium, C, sodium, D, lithium, E, magnesium. Quickly. Calcium, potassium, sodium, lithium, magnesium. Come on, guys. Let's finish this thing and do bone. I need to do bone. I must do bone this night. The sarcolemma of a resting muscle fiber is most permeable to what? What happened? Why is this question difficult? Why are you guys slowing down? Or you want me to stop? Trust me, I will. If you don't know the answer, type anything. All right. But I need to be seeing people typing. You understand? If you don't type, and maybe just 5 people, it's not encouraging, guys. I need to see everybody typing. If you don't know the answer, just type not sure 911 or whatever. That's fine. But you need to type something. Don't sit on the fence, please. Yes, it's potassium. Anytime you hear resting, just choose potassium. All right. Anytime you hear resting. You see the question, the sarcolemma of a resting muscle fiber. Just choose potassium. You'll be fine. All right. If it becomes active, that's sodium. Look at this question. Which of the following is true regarding the sarcoplasmic reticulum? I want to put the options now. I'm putting the options in the. This is A. But I need everybody to answer. If you don't know what to answer, just write anything. The truth of the matter is that the more questions you are exposed to, the better for you. That's the truth. This is D, and this is E. Which of the following is true regarding the sarcoplasmic reticulum? It stores the sodium needed for muscle contraction. B, it synthesizes and stores ATP. C, it helps to maintain the resting membrane potential. D, it releases acetylcholine into the synaptic cleft. E, it releases calcium when a muscle fiber is excited. Answer. Not sure. Good. Come on, guys. A, E. Continue. Let's go. Which of the following is true regarding the sarcoplasmic reticulum? Come on, let's go, guys. Two more people. Two. Keep going. We have up to 40 people here. It seems muzzle is the weakest point to honestly, like later go and watch this muzzle part very, very well. Fast forward to wherever we are right now. We spent almost 4 hours, 24 minutes. Fast forward there and watch the muzzle cells very well. All right, please, or make sure you join tomorrow. Maybe tomorrow I'll just start with muzzle. Probably that's what I probably have to do. And if I don't finish it, I'll tell them to go and watch this particular review. Okay, so look at guys. We say which of the following is true regarding sarcoplasmic reticulum? It has nothing to do with sodium. All right, it's calcium. It stores calcium. All right. So if you look at the answer, you just get your answer straight. Once you see calcium there, that has to be the answer. It releases calcium when muscle fiber is excited. Remember, the action potential comes down. You understand? And when in the T tube, right? And what happens? The cisterns, that's the ending part of the sarcoplasmic reticulum, contains what? Calcium. And the calcium goes in to cause contraction. Nine one one. I've explained it. Do you want me to re-explain it? If you understand it, I proceed. Someone say 911. Do you want me to re-explain, or I should proceed? Okay, I have to proceed because you are not talking, so that I don't delay others. Each T tubule is flanked by two dash. A bands, B, I bands, C, terminal cisterns, C, thick myofilaments, D, thin myofilaments. Answer. Oh, you want me to explain? Okay. I said that question was talking about which of the following is true regarding sarcoplasmic reticulum. You remember what sarcoplasmic reticulum is? Sarcoplasmic reticulum is the you understand the terminal part of the it's just like the endoplasmic reticulum, all right? But this time around, what it does is not protein synthesis. What it does is to store calcium. You understand? So when action potential enters inside like this, the ending part of the two two sarcoplasmic reticulum is the T tubule, right? Or is this two cisterns? In between the two cisterns, you will see a T tubule. So when action potential is generated, right, in the cell membrane, that's where action potential is generated, in plasma membrane. That action potential will pass the surface and enter into the cell through the T tubule. Now, when it enters it, as it's entering through the tube, entering through the T tubule, right, the calcium will spread inside and cause contraction. Remember, I told you the calcium holds the troponin, moves the troponin, which is the handle of the door. The door is the tropomyosin. You understand? Which is covering the active site of what? Actin. So calcium holds the troponin, which covers which is controlling the what? The tropomyosin. Then it moves the tropomyosin from the active site of actin, and myosin laps on top of it to cause what? Contraction. So that's what they're asking you there. So if you see the question, which of the following is true regarding the sarcoplasmic reticulum? The answer is that sarcoplasmic reticulum releases calcium when a muscle fiber is what? Excited. So the calcium goes and causes contraction. So this other question, each T tubule is flanked by two words. Answer is what? Terminal cisterns. You are very correct. The answer is terminal cisterns. All right. Very fast. This question, yes, terminal cisterns. This question I'm giving is the questions that I expect very fast response. Which of the following marks the boundaries of a sarcomere? Quickly, guys, quickly. I need 10 people to respond for me to be able to post another question. If you can do it very fast, it's good for all of us. Which of the following contains overlapping thick and thin filaments? Sorry, what am I even saying? Which of the following marks the boundaries of a sarcomere? Yes, Z disc. All right, that's the correct thing. Now look at this question very fast. We need to be very fast. Which of the following contains overlapping thick and thin filaments? Quickly, 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 quickly. Overlapping light and dark band. Overlapping thick and thin filaments is what? Two more people. Come on, guys. Come on, come on, come on, come on, come on. Don't sleep. Why are you sleeping now? I'm smelling that somebody is sleeping. Some group of people are sleeping here. Wake up. You want me to go and sleep? Yeah, I like to sleep. Oh. Not sure. Yeah, so you need to go look at that picture very, very, very well. The muzzle picture. You understand? Look at the muzzle picture. Extremely important that you look at that picture because, like, let me show you. Where is muzzle? Is this muzzle? Yes. No, this is bone. This is muzzle. You see this diagram? You see the diagram? You need to study this diagram properly. All right. You need to study this diagram extremely. This is Z disc. This is another Z disc. The distance between this Z disc and this Z disc is what? Sarcomere, right? Now, this Z disc is attached left and right by what? By the thin filament. That's the actin. All right, flanked by the titin. The green part is the titin. All right, titin up, titin down. But the orange part is the actin. So this is the thin filament. So this thin filament to this thin filament is I band, right? But you see, this I band now overlaps with this dark band here, or this thick filament and here. So between here that they started overlap to here that they stopped overlapping is called what? A band. You need to know what A band is and what I band is. Extremely important. You can see this picture and they will point to something and they will tell you to answer. You have to answer. Do you understand? And this area where you see only the dark band is called what? Only the dark band is called what? H band. And the middle of that H band is the M line. All right? This thing is extremely important. All right, look at this question. Look at this question here. Driving to work. Oh, that's fair. Look at this question, guys. In skeletal muscle, the term that describes the alternating light and dark band is what? A. Strabismus. B. Striation. C. Tetanus. D. Myofibris. E. Myofilaments. Myoblasts or whatever. Yes, the answer is striations. And look at this. I'll give you just couple of maybe 5 more questions, and that will be all for this thing. Then I will encourage you to go and look at the muscle video, please. The protein that acts as a calcium receptor in skeletal muscle is what? Yes, it's troponin. What's the answer to this? Exactly, the answer is troponin. The answer is troponin. Look at this. Yes, the answer is troponin. Thank you so much. You guys are coming back to life. How many somatic motor neurons stimulate one muscle fiber? Quickly. Quickly. 2 more people to answer and I'll move. Yes, the answer is 1. All right. Look at this question. The process of engaging, yes, the answer is 1. The process of engaging more motor units to increase the strength of a muscle contraction is called what? It's called what? A, wave summation. B, recruitment. C, temporal summation. D, incomplete tetanus. C, E, complete tetanus. Answer. Yes, you guys are correct. The answer is 1. And the other one is troponin. Look at this question. To stimulate muscle contraction, acetylcholine is released from the dash into the synaptic cleft. Where is acetylcholine released from? Yes, that answer is recruitment. Yes, the answer is recruitment. You're correct. The answer is recruitment. That's B. Yes, you're correct. Look at this question. To stimulate muscle contraction, acetylcholine is released from the dash into the synaptic cleft. A, synaptic vesicles. B, postsynaptic membrane folds. C, sarcoplasmic reticulum. D, sarcolemma. And E, terminal systems. Where is acetylcholine released from? That pours it into the synaptic cleft. Quickly. Through exocytosis. Where? Exactly. The answer is vesicle. From the vesicle. Look at this question. I need to be sure that you guys understand this thing properly so that anywhere they ask question is midterm. Question can come from anywhere. Anywhere they ask question, you'll be able to answer it authoritatively. The axon terminal releases acetylcholine into the synaptic cleft, which is received by the receptors on the motor end plate. Which structure does this describe? A, the sarcomere. B, the thick filament. C, the varicosity of a smooth muscle cell. D, the neuromuscular junction of a skeletal muscle. And E, a motor unit. Answer. I'll take the options again. A sarcomere. B, the thick filaments. C, the varicosity of a smooth muscle cell. D, the neuromuscular junction of a skeletal muscle. And E, a motor unit. Answer. Okay, I'll ask you 3 more questions and that will be it for muscle, okay? All right? Then maybe we'll take some questions in bones and we'll call it a day. Because we've tried. The other ones, genetics, I have videos for them. I have videos and I did review with you. So go and look at that review. That's fine. Or tomorrow, you come again and, you can join. Tomorrow, I'm going to start from muscle, bone, downwards. All right? I know you guys are tired already. You guys have really tried, honestly. Yes, the answer is neuromuscular junction. The answer is neuromuscular junction. So I'm asking 3 more questions and that will be it for muscle. 3 more questions. Look at this. Which of the following best describes the resting membrane potential? Which one best describes it? The RMP. Let me give you the option. I'll give you 3 options. This 1A. This is 2. 2. That's B. This is C. Okay, I say I will give only 3. It's okay. Let me stop it at 4. Which of the following best describes the resting membrane potential? A, the intracellular environment is negatively charged. B, the intracellular environment has more positively charged sodium. C, the extracellular environment is negatively charged. D, it has a voltage of about plus 75 MV. And E, it is dependent upon the absorption of sodium, potassium ions by the muscle fiber. Whatever. Oh yeah, choose. Very fast. After these, 2 more to go. So what is the answer? You guys are very correct. The answer is A. It's negatively charged. Every other thing is not correct in that question. Oh yeah, look at this question. Sorry, I won't be able to be explaining a lot because I really want to hurry up. Which statement best describes the goal of medication used to treat myasthenia gravis? Which? Okay, it's true. I have to put the options, right? Yeah. I'm just gonna put a few options. A. Anyway, let me just put everything in case if you mistakenly see the question. B. C. Yeah, I know you're answering the other question. Yeah, that's fine. You're correct. D. E. After this one more to go. Answer. Which statement best describes the goal of medication used to treat myasthenia gravis? To increase the number of acetylcholine receptors, to decrease the number of synaptic vesicles, to promote multiple motor unit summation, to inhibit calcium release from the sarcoplasmic reticulum, to inhibit the function of cholinesterase. Answer, guys. Come on, let's go. You guys are asking somebody. Okay. Yes, the answer is E, to inhibit the function of cholinesterase. The answer is E. Please try to look it up. The answer is E, to inhibit the function of cholinesterase. Look at this one. That's the final question for muscle. During muscle contraction, binding sites for myosin are uncovered by the movement of dash. What uncovers the binding site? Binding sites. What uncovers it? That's the active site of actin. What uncovers it? During the contraction, binding sites for myosin are uncovered by the movement of dash. You see, this is why illustrations are very, very good. I really like what you just said now. It's extremely good because you thought about it. I just have to open the door, right? Yeah. And it is not the handle that is covering the door. That is covering that space. It is the door itself. So it's tropomyosin. You guys are very correct. They can also ask you during muscle contraction, where does calcium bind? You know, calcium binds to the handle. Nobody can forget this one. All right. Okay. So that is muscle. If you guys are tired, Then maybe you can join us for bone tomorrow. Or do you want me to continue? 10 people, we vote. If you want me to stop here, I can stop here. If you want me to still go for the bones, I'll go for the bones. Somebody say, hey, sorry, it's almost 8 chapters. What are you doing? It's possible that you can get equal questions in each or not. I don't know. Depends on what the spirit ministers onto your professor. You say I can never start. No. I just need. The thing is that I like it when people are very active with me, right? But it seems the activities level is decreasing. So I need votes. 10 people. Same wasn't a vote. Didn't settle into my spirit. Oh, sorry. I could have. Okay, what will happen? Like tomorrow, I'm going to discuss the muzzle slide before I enter the question. You understand? That's so. If you are not very confident about it, you can just join tomorrow. Maybe when I do muzzle bone, you can go off. All right. But I will start with tomorrow. I'm going to start with muzzle bone. You understand? Though I wanted to start with cell because that's what we've done a long time, just to remind you guys. But it seems you guys already have the grip of it, which is fair because you've written the exam on it already. So let's vote. 10 people. Yes, I continue. No, I stop. Tomorrow I continue. No, I can't do bone in 15 minutes. It's not even possible. It's not even possible. You've not been tested on bone, so I work tomorrow from 7 p.m. You can look at the video now. That's fine. That's fine. So I have 1 yes, 2 no's. No, tomorrow I'm starting with muscle, then after that bone. So that people that, because if I start with muscle tomorrow, bone, even if I don't finish, I can tell them to come and watch this. All right? Yeah. Then some of you that, if maybe we are not able to finish, you can just jump into the other one. What time tomorrow? Oh, tomorrow from, I have class till 9. But tomorrow I'll start from maybe 8. Between 8, 8:30. I'll send a message. Okay, somebody said I should stop. He said, Nchebe, you've tried a lot. You've suffered for us. I should stop. Somebody said yes. We have 3 yes. We have 4 no's. 4 no's. Start at bone tomorrow to continue where we left. No. I will start at muscle tomorrow. I'll review muscles tomorrow. Muscles and bone, because a lot of people seems to have more challenges in muscles and bone. So I'll review it tomorrow. So if you're able to join, fine. But if you're not able to join, you can easily just go on live. You understand? Maybe on Tuesday morning, go on live and look at the first 1 or 2 hours of the this thing, because it will be muscle and bone. Yeah, tomorrow 8:30. Yes, depends on when I'm back. If I come back before 8, then I'll start 8. If I don't come back before 8, it will be 8:30. But I know I will come back before one of those times. Then continue now. See face. And also another thing is that I'm releasing muscle bone video tomorrow. I might do it this night. If I leave you guys, maybe I'll do it this night, maybe before 3 a.m. Or I'll just sleep, wake up tomorrow morning and do it before going out. You understand? So I think that will work. I think we can actually stop here. Then tomorrow, let's take care of that bone. I'll post you the video. So even if you are not joining tomorrow evening, for people that don't have time, you can just watch the video. How many more chapters to go? There are a lot of, the thing is that, let me see. So the thing is that we were supposed to finish everything today, but genetics, I've already done genetics and you have a video for it. I'm going to post the review we did on genetics and you have a video for it. I think so, yes. This one is tissues. I did a review with you on videos and I posted it, so you can actually check that up. Skin, the same thing. So the only one that is left is more like bone and tissue, and the muscle, which we just finished muscle now. So it's just bone. Yeah, just bone. So what seems a lot of people want us to stop now. So what will happen is I'm going to post that. Please, if you've not subscribed, subscribe and click notification so that when I post it, even if I don't remember to post it on Canva, you can just see it. You will see it as notification on your phone. All right. And the idea, and also, when you will be doing this should be one. When you'll be doing AP2, I may not be the one that is lecturing you, but when you have your notification on, you'll be able to see that I am on live and you can just come in. All right. It will help you a lot. It will really help you a lot, even if I'm not the one that's teaching you. So let's stop here. We've tried. We went almost 4 hours, 50 minutes. Right? 4 hours, 50 minutes. Oh, I have dimple. Oh, because I'm losing weight, my dimple started coming out. I used to have dimple, but it's unilateral one. Very deep. Very, very, very deep. Yeah, it's the same info. So, but tomorrow we start with bone and muscle. So even if you are not with us tomorrow, just come in for bone and muscle and you can just go. All right. And I'll post a video. Make sure you watch that video. I'll post a video today. And another thing you can do is, since you have part of this review, you can also check all the videos that I've posted for you. It seriously will help you. Listen, no matter, even if you've seen those questions before, the more you attempt it, the more you listen to it, the better for you. All right. So you can clear your midterm, because midterm is raw mark. Honestly, midterm is raw mark. When they say something is raw. I know where your mind is going to, but that's not what I'm talking about. I'm talking about the term, the mark. It's raw mark. All right. So that's why I will encourage you to make time out to see those videos as much as possible. Just listen to my ugly voice talking. All right. That's fair. Yeah. So, okay. So that's it. I will. This is 11:50. People are going to work the morning. I will leave you guys to be. All right. So tomorrow, try to. I will send a message if I remember. But if I don't remember, put your notification on so that I can, get to the information. And make sure you subscribe if you've not subscribed. All right. You guys are the best. Thank you so much. Yeah. I don't want anybody to have reason. All right. I don't want somebody to tell me, hey, Professor, I didn't do this. No reason. No 100. No reason. No reason. Yes. No reason not to get all your questions, not to get 100 over 100. There is no reason. I'm telling you. All right. Because I bet you're going to see questions that you've seen many times, several times. You understand? So I don't want you to, overthink questions. Don't overthink. Don't have anxiety. Don't overthink. Just answer your questions, attempt your questions, and move. All right? Yeah. Don't think too much about it. Don't think too much about it, please. Because that's just like they asked somebody, the minimum stimulus needed to cause muscle contraction is called DASH. Threshold, latent period, twitch, motor units, innervation. This is thinking twitch can be the answer. Motor units, maybe. When something you can just pick threshold and move. Do you understand? Like, don't overthink answer. You know it already. Don't overthink it. All right? So you guys are the best. Thank you so much for being with me. We still have up to 32 people online. That's great. Thank you so much for being with me. And if you miss out anything, try to come back to the channel and see the video. All right? Don't forget. Try to come back and see the video. I'll just post a link of this to your canvas so that you can easily just click there and watch the video again. If you need to fast forward to a certain topic that you didn't get properly, please fast forward. All right? You're welcome. Thank you so much. So see you guys later. Thank you. Thank you so much. You guys are the best. Take care of yourself. You're welcome. Thank you. I will not forget. Say chicken. Ah! Hey, God, no. Somebody said my laugh is terrible. I said, it's a pity. That's how I laugh. Somebody said my laugh is funny. Yeah. Thank you very much. You guys are great, man. Take care of yourself. Yeah, thank you. Thank you. See you tomorrow. Take care. Don't forget to comment later. When you are watching the video, you can also still leave the comments, still leave a like, and still follow the channel. All right. So I'm going to end the stream right now. You're welcome. Take care. I'm waiting for you to stop it in case I miss something. You are very funny. Oh, wow. I'm actually slimming down. See, it's actually going inside when I laugh. I'm actually slimming down. Ah, interesting. So this, my oatmeal is working. It's actually working. Thank you so much. Thank you so much. Okay, you want me to end the stream? Okay, let me end the stream now.